So we're going to be looking at both the nervous system and also endocrine systems, and also the broader topic of homeostasis, and a couple of examples of homeostasis, things like the control of blood sugar and also osmoregulation in the renal system. Now again, in keeping with the format of the previous webinars, there's an awful lot to get through in this particular topic. Now, if we don't manage to get through all of the materials in the 3-hour time limit, what I will do is I will leave it open to the students who are in attendance as to whether they want me to continue or whether they'd prefer me to end the webinar, do the Q&A, and then upload an additional video or schedule an additional webinar to cover the topics that we don't manage to cover in this webinar. However, this topic is an enormous topic. It contains an enormous amount of detail. So what I've done, just before we begin, so that you guys are aware, the chances are that we probably will not get to cover absolutely everything in this topic, even if I do have to film another video to cover it. So what I've done, I go to the website here, I have actually made there's a couple of lessons which I have made free lessons in the course module which covers this topic. So if you go into the course modules here, and you don't have to sign up to access these lessons. If you go into this module here, the control systems topic for advanced level, you'll find that there are 2 lessons which I've made freely accessible to anybody, whether or not they're enrolled on that course module, or whether or not they're a student who has an account on the site. The first is on synaptic transmission. The second is on muscle physiology. Now the synaptic transmission lesson video is about 22 minutes long, and there's also you can also access the lesson slides that I'm going to be going through in that video, and there's also an end of lesson quiz which you can access, and all you need to do to access that is to register a free account on the website. There's also another lesson on muscle physiology. Now the section of the AQA specification that deals with muscle physiology is deceptively short, but the actual amount of detail that you need to know about in order to fully satisfy the learning outcomes in that section of the specification is actually very, very large. So there is quite an in-depth lesson video here, which is about an hour and a half long. Now you can access that at this page on my website. Alternatively, what I've done, and again there is an end of lesson quiz and also access to all of the PowerPoint slides that I'm going to be going through in this in that lesson. But alternatively, you can also go to the YouTube channel for my website, and you'll find that I've uploaded that lesson on muscle physiology to the website. So you can also find the other free lesson videos here as well. So there's a couple of lesson videos here that relate to this topic, muscle physiology here, and also synaptic transmission. And so in the interests of trying to keep to the 3-hour limit, I won't be going over the muscle physiology topic in this webinar, and I won't be going over the synaptic transmission topic in this webinar, just because if I did, then the webinar would probably stretch to about 6 hours. Now in the control systems module, there's around about 9 hours worth of teaching videos here, and hundreds of quiz questions and lots of other resources there as well. So if you are interested, please do have a look at that course module and consider enrolling there. I realize that many of you have already enrolled on that course module, and also the students at the SWAP consortium have already been given access to that. Now I have cleared that with those of you in the private Zoom room. You're all aware of that, but just so anybody watching on the YouTube stream, if you're wondering why I don't go over the synaptic transmission or muscle physiology topics in this webinar, it's because those are already available to you in these teaching videos on the YouTube channel. So I'd encourage you, if you have any questions on those, obviously those of you in attendance, you can simply type those questions as they occur to you into the Q&A box, and I'll address those at the end of the webinar. Alternatively, you can go to the YouTube pages for these videos and post your questions as comments there, and I'll answer your questions there. And finally, you can always go into the site, go into whichever of these lessons you want to ask questions about, and type your questions here in the comments, and I will respond to you there. And obviously those of you who are already private students enrolled on this course module, you will have your own private coaching area where you can interact with me privately, ask questions, upload work that you need help on, and also get private academic coaching on this topic as well. So with that all out of the way, let's get started then. So the first thing that we're going to cover is the first section that appears on the AQA spec in this topic, which is on the subject of the control of plant growth. So, from an early age, we learned that seedlings will grow towards the light, but what controls the growth of seedlings towards the light? Well, it's the light itself that controls the... Is it the light itself that controls the growth, or does the light control some other factor, such as a chemical growth factor? Now, it turns out that if you direct the light towards the plants from different directions, as you see here with these mustard seedlings, you'll find that they tend to grow towards the light wherever the light seems to be coming from. So it can be shown that plants respond to light and that they use growth factors, chemical substances that control the growth of their shoots, and it's these growth factors and the ways in which they operate within the shoots which controls the directions that those plants will grow towards in the case of light. So, now what controls the growth of the roots downwards into the soil? Well, is it gravity, or is it the absence of light that controls the growth, or is some other explanation involved, such as some chemical growth factor? Well, again, it can be shown that plants respond to gravity, and that the growth factors which are present in those plants, the chemical substances which we call growth factors, are responsible for controlling the direction of the growth of the roots downwards into the soil. So plant growth factors are chemical substances which are produced in plants which accelerate, inhibit, or otherwise will modify or regulate the growth of different parts of the plant. So in order to do that, they can affect cell division, cell elongation, and also cell differentiation, and they also control activities such as the growth of the roots and shoots, the development of flowers, and the ripening of fruits, and also the falling of leaves and fruits. So we can refer to different types of growth behaviours called tropisms. So plants respond to a very wide range of different stimuli which are present in their environments. Now, unlike animals, plants generally cannot undergo locomotion. They cannot undergo movement from one place to another. So instead, their response occurs by means of growth. So a tropism is defined as a response to a directional stimulus. Now, the type of growth response is named by prefixing the word tropism with the first few letters of the type of stimulus which is responsible for that tropism. So, for example, we can say that phototropism is a response to light, hydrotropism is a response to the presence of water, gravitropism is a response to gravity, thigmotropism is a response to touch. Now, these are all growth responses that plants will exhibit to these different types of stimuli. But we can also define further each of these tropisms as either being positive or negative. Now, if the growth response is towards the stimulus, the response is described as being a positive tropic response, and if the growth is away from the stimulus, it's described as a negative tropism. So, for example, we can say that shoots are positively phototropic, and that roots are negatively phototropic. However, we can also say that shoots are negatively gravitropic because they grow in the opposite direction to the pull of gravity. And similarly, we can say that roots are positively gravitropic because they grow towards the influence of gravity. So, phototropism can be easily demonstrated in things like broad bean seeds by setting up three different conditions, and we see that in this diagram here. So we see here that we have three different broad bean seeds, and we are growing them or germinating them under differing conditions. So for seedling A, we are directing the light directly downwards towards the seedling. Seedling B is grown in the dark, and seedling C is grown under conditions where the light is being directed towards the seedling laterally, like this. So, in the case of A, this is a plant grown outdoors. B, the plant grows in the dark. We can see develops a long, thin, spindly stem and also yellow leaves. And this condition, where the plant exhibits these symptoms, is called etiolation. And here in experiment C, we can see that the plant which is illuminated from the side curves towards where the light is coming from. Now, we can use a device called a clinostat to demonstrate phototropism. So we can have a plant inside a box, and there's a slit in the side of the box to allow light to come into the box from the side here, and we can see that the plant grown under these conditions will curve towards the light, towards where the light is coming from in this slit in the side of the box. But we can also put a similar plant in a similar box, but this time place it in a clinostat. Now a clinostat is a very simple device. It's basically a device which allows you to rotate the plant pot continuously, and also to angle the plant pot at a particular angle as well if that's required. So if we place it on a clinostat and then rotate that clinostat continuously, even though the plant is receiving the light from this direction, we'll find that the plant grows straight upwards. So we can do a number of experiments with plants and to find the influence of different growth factors using a type of hollow protective sheath that grows around the stem apex of the plant called a coleoptile. So this diagram here shows a germinating seed. Here we see the initial flag leaf which emerges from the seed as it germinates, and that's flag leaf, which is the first foliage and is rolled up, is surrounded by a protective sheath called a coleoptile. So a lot of experimental work can be done with plant growth factors, and this often relies on exogenous application of the substances on the excised coleoptiles. So this covers the whole of the emergent part of the seedling, and it persists until the first leaf pushes its way through the top. So let's have a look at some of these experiments then. So here we see an investigation which investigates the effect of covering different parts of the shoot tips of 3 wheat seedlings that we see here. So in experiment A, we see that the tip of the shoot tip is covered. In experiment B, the shaft is covered, and experiment C is the control, which is uncovered. So when we expose these seedlings to light, which is coming from a lateral direction like this, we find the following result. So we can see here that the seedlings B and C are growing towards the light, but that the seedling C is bending all the way along its length towards the light, whereas seedling B is only bending in the areas which are exposed to the light, and seedling A is unaffected. So we can so essentially these are the questions which this experiment is intended to answer. Let me just go back one there so you can see that. So we can do a further experiment which investigates the effect of removing and then replacing the shoot tip of a wheat seedling. So we take the tip of the growing coleoptile and we find that the growth stops. If we replace the tip, we find that the growth is resumed. So another experiment investigates the effect of replacing the shoot tip of a wheat seedling. So here we have the tip which has been sliced off, but it's been separated from the rest of the coleoptile by an impermeable sheet of mica. Now under these circumstances we find that the growth stops. If we then replace that sheet of impermeable mica with a permeable block of agar jelly, we find that the shoot tip continues to grow. So a growth promoting substance, the plant growth factor indole acetic acid, or IAA as an abbreviation, we know from these results that this diffuses out of the shoot tip and is and then down through the agar jelly, and it's this chemical that is generated in the shoot tip and diffuses down into the growing shoot, which is responsible for the elongation of the shoot, the growth of the shoot. So IAA is also known as auxin. So this experiment demonstrates that IAA can be collected directly from the shoot tip of a wheat seedling, and that it's the auxin, the IAA, that directly controls the shoot growth. So we take the tip cut off and replaced onto an agar block, and then we discard the tip, and we take the agar block and place it centrally onto the coleoptile stump, like this. Now when we do that, we find that the growth then continues. So we've demonstrated that the plant growth factor, indole acetic acid, or auxin, is responsible for the growth of the shoot, but we've also demonstrated that this chemical is being generated in the shoot tip. So this next experiment demonstrates that the auxin isolated from a shoot tip can cause the curvature of the shoot tip of a wheat seedling. So again, we take the tip off, we place it on a permeable agar block. After a short while, we discard the tip, and then we take the agar block, but this time we place it asymmetrically onto the coleoptile stump like this. Rather than placing it right the way over the stump, we place it on one side of the stump only. Now when we do this, we observe that the stump will curve in the direction opposite to where we placed the agar. So we can also demonstrate that IAA is redistributed, and we can also explore the role of light in this process. So in this experiment, we start with the coleoptile tip removed and then illuminated from one side for a period of time. We then place that tip onto two agar blocks in the dark. Now these agar blocks are themselves separated by a strip of impermeable mica. We then place the agar blocks again asymmetrically onto two coleoptile stumps like this, and then we find that the greatest curvature is produced from by the block from the dark side. So again, if we are illuminating the coleoptile tip from this direction, then this is the agar stump from the dark side of that coleoptile tip, whereas this is the agar from the illuminated side. So we find that the agar block from the dark side shows more curvature than the agar block from the illuminated side. So from this we can conclude that light causes more auxin to gather on the side of the coleoptile or the coleoptile tip furthest from the light, the shaded side. Therefore, more IAA will pass down the shaded side of the shoot, causing the greatest curvature. So, light causes the redistribution of auxin and not its inactivation. So, here we can demonstrate again in a slightly different experiment that IAA is redistributed. We have two different seedlings here, A and B. Seedling A is kept in the dark, and seedling B is kept in unilateral light, light which is coming from one lateral direction here. So we then extract the IAA from the coleoptiles in both cases, and in both cases the amount of IAA is about the same. So we see here that whether light is present or not, the total amount of auxin that's produced and collected is about the same. Light does not therefore inhibit auxin production, and it doesn't subsequently destroy it either. What it does is it influences the distribution of that growth factor. So seedlings can have a thin glass plate inserted into their tips like this, and obviously glass is an impermeable material. The seedlings are then oriented so that the glass plate separates the two sides of the coleoptile, one side being illuminated and one side being in shade. So we can then extract the IAA from the coleoptiles, and in both cases the total amount of IAA is about the same from both sides of the plate glass. So while the plate glass does not affect the transmission of light because it is a transparent material, it does prevent the IAA being transferred from one side to another. So again, we see that from this experiment we can conclude that light doesn't destroy auxin, it doesn't promote the production of more auxin. What it does is it causes the distribution, redistribution of auxin, but when we prevent that from happening by separating the two sides, the illuminated side and the shaded side, using this glass plate, that redistribution is no longer possible, and we find that the auxin concentration on both sides is approximately the same. So if the concentrations remain equal on either side of the plate, we see that no bending occurs during the growth of this shoot. So the role of IAA, well, auxin is produced in the apical meristem of the shoot, of a shoot or a stem. In unilateral light, in other words, under conditions where the plant is being illuminated from one side, the auxin is redistributed. It moves over to the shaded side. Now the shaded side develops a higher concentration of IAA than the illuminated side because of that. And then the IAA stimulates cell elongation and division, and therefore the cells on the shaded side will divide and elongate more than the cells on the illuminated side. And it's this unequal growth that causes the shoot to, or the stem, to bend towards the light. So, we've talked about phototropism. Let's have a look at some experiments where we can demonstrate gravitropism. Now again, gravitropism is the growth movement or the growth behavior of plants in response to the pull of gravity. So here we see a number of different seedlings here. In seedling 1, we place a bean seedling in a horizontal position in the dark with markings made on both the growing shoot and the growing root at 1 millimeter intervals here. Next, we find that under conditions where we allow this seedling to grow, we find that the shoot develops upwards. In other words, it bends and curves upwards. So we say that the shoot is showing negatively gravitropic curvature here, and we can see that the markings that we made are starting to move apart as those sections of tissue undergo cell elongation and cell division. Now, in contrast to that, we see that the root is showing positively gravitropic curvature, and again, the markings indicate where the cell elongation occurs. Now, if we then rotate this seedling 90 degrees to this orientation, we find that the old growth curvature is unchanged, but new growth curvature develops only behind the root and the shoot tips. So what is the mechanism of gravitropism? Well, gravity is thought to cause, again, a redistribution of indole acetic acid, or auxin, in both the root and the shoot in seedlings which are grown in a horizontal position. So the IAA will gather here and cause accelerated cell elongation, and this in turn causes a curvature upwards. And also the IAA will also gather here and will retard the cell elongation in the case of the root tip, causing a downward curvature. So we can say that auxin accelerates elongation in shoots but retards cell elongation in the roots. So roots, for this reason, are positively gravitropic and shoots are negatively gravitropic. So again, we can use a clinostat to demonstrate this principle. So when we set the clinostat to rotate once per hour, the force of gravity, if we orient the seedling in this orientation, the force of gravity acts equally on all parts of the seedling over that given period of time. So as the seedling does not remain in any one position long enough to make a gravitropic response, we see that both the root and the shoot will grow outwards horizontally. So this is the setup for that experiment, and again, a clinostat is simply an electric motor with a platform on which you can mount a growing plant to investigate the effect of different orientations and rotation on gravitropic and phototropic behaviors. So to summarize then, the mechanism of IAA action in the phototropic response of a shoot and a root is shown here. So the IAA will accumulate on the shaded side. So when unilateral light is shone in one direction towards the shoot tip, the IAA will redistribute itself so that it is at a higher concentration on the shaded side. Here it will stimulate growth in terms of cell division and cell elongation, and this will cause curvature on that side at a greater rate than the curvature on this side, and therefore the shoot will bend towards the light, causing a positively phototropic response. Now in the roots, the IAA has the opposite effect. So if we illuminate the roots in a unilateral direction, like this, we find again that the IAA will accumulate on the shaded side, but in the case of the roots, it inhibits growth. It slows down cell elongation and cell division so that this side will elongate faster than the shaded side, and in this way it will cause a negatively phototropic response. So the mechanism of action of auxin in the gravitropic response of a shoot and a root is shown here. So we have here a shoot oriented horizontally, and so gravity is working in this direction here. Now under these circumstances, the IAA will accumulate on the underside of the shoot, therefore it will cause more rapid cell elongation and cell division on this side than it does on this side, and this will cause a negatively gravitropic curvature and response, causing the shoot to grow away from gravity. Now, if we contrast that with the root, the IAA will accumulate on the underside, but here it causes a positively gravitropic response. Apologies for the noises in the background, guys. It's again a very hot day here, so I'm forced to open the windows, so I hope the background noise doesn't become too distracting for you. Do let me know if that is the case, then I'll see if I can do something about that. So high indole acetic acid in the roots has the opposite effect that it does in the shoots. It slows down the growth. It retards the rate of cell elongation and cell division, and this causes a positive gravitropic response where the root will curve downwards in its growth towards the direction of gravity. Okay, so the next section that we need to look at is to do with innate behaviours, and these include taxes, kineses and reflexes. So what are taxes and kineses? Well, these are simple instinctive forms of behaviour or innate behaviours, and these rely almost entirely on simple reflex actions. Now, these are forms of behaviour involving the movement of the entire organism from one place to another, and we refer to this as locomotion. Now, the organisms will do this in response to a specific external stimulus. So the adaptive value of taxes and kineses is their role in helping to maintain the organism in the most favourable environment for their survival. So a taxis or a tactic response is defined as the movement of a whole organism or cell in response to a directional stimulus or the direction of a stimulus. So certain organisms will move directly towards or away from a specific stimulus. So in other words, they will show either a positive towards taxis or a negative away from taxis. Now, taxes are also classified in accordance with the nature of the stimulus. So movement of an organism directly towards a light source, for example, is described as a positive phototaxis. Now, there are other taxes, including chemotaxis, hydrotaxis and aerotaxis. So for example, silkworm moths, like this chap here, the males of this species will display a positive chemotaxis when they fly directly towards the chemical secreted from the abdominal regions of the female moth. Now, this chemical bombicol is picked up by the feathery antennae that you see here on the male, and the concentration gradient will guide the male towards the female, because obviously the further away the male is from the female, the lower the concentration of the bombicol molecules in the atmosphere, but as the male moves towards the female, that concentration will increase. So these antennae will allows this organism to show a positive chemotaxis towards this chemical. So water fleas, or Daphnia, like this chap over here, these have two instinctive behaviours, a phototaxis and an aerotaxis, and this causes them to swim near the surface of a pond where the light intensity is highest, and also the dissolved air or the dissolved oxygen concentration in the water is also at its greatest. So this Daphnia will swim directly towards low intensity sunlight, so it displays a positive phototaxis, but it also swims directly towards the oxygen rich environments in the water, and again this would for this we would say that it displays a positive aerotaxis. Maggots will also show phototaxis. In this case they will move away from the light, so we say that they display a negative phototaxis. So if we perform a simple choice chamber experiment like this, where we introduce a maggot into this chamber, and when it can only really move in this direction, when it reaches the fork here, the T-junction, it can either move into the illuminated section here or into the dark section, which has been covered by a piece of card preventing the light from getting in. We find that it typically will choose, more often than not, to move towards the darkened section, the dark arm of the choice chamber here. Now, observations show that when the maggot enters the dark arm, it will stop and stay there. However, if it enters the illuminated arm, it will start to move up, and it will continue to move up and down, and inevitably it will find its way back into the black or the darkened arm of this T-junction here. So, a kinetic response, or a kinesis, this is a non-directional response in which the rate of movement is related to the intensity of the stimulus, but not its direction. Now, pay close attention to this wording here, the rate of movement, not the direction of movement, but the rate of movement. So really what we're talking about here is the speed of movement, how fast it's moving or how much distance it's traveling within a specific period of time. So there are two types of kineses. There are orthokineses, in which the rate of movement changes in response to a stimulus, but there are also clinokineses, in which the rate of turning changes in response to a stimulus. Both of these are classified as kinetic behavioral responses, or kineses. Now, one of the most common organisms which are used in school laboratories to demonstrate these types of innate behaviors are woodlice. So these belong to the phylum Arthropoda and the class Crustacea and the order Isopoda. Now, these are terrestrial species and they lack many of the water-conserving adaptations found among the other members of their phylum. So for that reason, and also because they have a relatively high surface area to volume ratio, they lack a waxy cuticle, they also have rather primitive respiratory surfaces, a pair of pleopod lungs, which are open to the atmosphere through a permanently open pore. Now, because of all of these different characteristics, this makes them particularly prone to drying out or to desiccating. So, for this reason, they show very clear patterns of behaviour which are aimed at limiting water loss and promoting their survival in a range of different terrestrial habitats. So, kinesis again is the movement of an organism in response to a stimulus where the speed of movement depends on the strength rather than the direction of the stimulus. Again, orthokinesis is when the speed of the movement of the individual is dependent upon the intensity of the stimulus. So a good example of this is the behaviour of woodlice in respect to changing humidities in their environment. So, woodlice will display kinetic behaviours when they encounter the atmospheres of different humidity. In drier environments, they tend to increase their rate of movement and decrease their rate of turning. So, students investigated the behaviour of woodlice when given a choice between dry and humid environments. 20 woodlice were introduced into a choice chamber that provided them with a choice between dry and humid atmospheres, and the distribution of woodlice was recorded after a period of 3 minutes, and then the investigation was repeated to include 6 trials. Now, this is a fairly common experiment, and you may well have done similar experiments like this for yourselves. So the choice chamber consisted of 2 halves with distilled water introduced into one side and a drying agent, calcium chloride, introduced into the other to provide a more humid atmosphere. Now, in this investigation, the tactic responses are being observed as the woodlice demonstrate their preference for a particular environment by moving towards or away from that stimulus, in other words, the level of humidity. So the results of this investigation, the 6 trials, are shown here. So at the end of each of those investigations, these are the counts of the number of woodlice found in the 2 halves of the choice chamber. So now obviously the analysis of these types of results is probably something that you will have done yourself in the course of doing practicals like this, and I believe all of the exam boards at advanced level require you to have done at least one practical where you are measuring the changes in the behaviour of an organism in response to a named environmental variable. So this is actually quite a common type of investigation. Now, for this type of investigation, generally you would need to analyse the results statistically. So you would need to state a null and alternative hypothesis for this investigation, and then analyse the results statistically using a chi-square test with Yates correlation. Now, I'm not going to go into that in too much depth here. However, if you are interested in the kind of the in-depth explanation as to how that's done, again, you can either go to the control systems course module, where we go into that in a bit more depth when we explore taxis, kineses and reflexes. But if you really are after the kind of in-depth teaching on the applications of statistics in advanced level biology, I'd advise you guys to have a look at this course module here, where we get into all of the different statistical tests in a lot of detail, and I also provide you with a number of pre-configured spreadsheets which you can use to analyze that data, and also to allow you to remember how to select the appropriate statistical test for a particular set of data. So another experiment can be done, which is, again, probably an experiment that many of you will have performed yourselves in the course of completing your advanced level biology course. So this experiment would be investigating orthokinesis in woodlice. So a group of students investigated the rate of movement of woodlice when subjected to environments of differing humidities. So we have here basically just a petri dish with a little porous platform here for the woodlice to walk around on, and in the bottom of the petri dish we have a glycerol water mixture. Now the proportions of the mixture can be varied to vary the level of humidity in the chamber. So different chambers like this of different humidities were prepared by preparing different proportions of glycerol and water mixtures, and then the distance moved by the woodlouse in a given time was determined. So 6 individual chambers of different humidities were set up, and these were the results. So these were the relative humidities at the different percentages of glycerol. So again, this is probably something that you may have performed yourself or something similar to this. Now a single woodlouse in each case is introduced via the entry hole, and then the rate of movement was determined over a period of 60 seconds. Now the way that you do this is by means of taking a marker pen and positioning that marker pen above where the woodlouse is, and then when you start the timer for the experiment, you simply follow the woodlouse around on the surface of the petri dish, and then you can then measure the length of that line after the experiment is concluded. So this is an example of a tracing from a low humidity chamber. And again, you can determine the length of this line reasonably accurately by placing the top of the petri dish over some squared graph paper like this after the experiment. Now again, this is the line that was traced by the woodlouse in a low humidity chamber. Now if we contrast that with the tracing produced in a high humidity chamber, we can see that there's an obvious difference. So we can calculate the mean rate of movement per minute in centimeters per minute here, and we see that the higher the humidity, the lower the mean rate of movement of the woodlouse. So again, you would need to take a number of different steps to analyze and interpret these results. Obviously, the first of those steps may well be that you present the results graphically, and the kind of conclusions that you can draw from this type of experiment would be to discuss the adaptive value to the organism of the observed responses to woodlouse survival. Now, it's a fairly obvious conclusion here. The higher the humidity, the less need the woodlouse has to move around to find a better condition for itself to prevent desiccation. And so typically, the higher the humidity, the lower the mean rate of movement. But again, there's much more detail on these experiments and also many others, together with how these results from these experiments are analysed and interpreted in both the control systems course module on my website and also the statistics course module there as well. So I'd encourage you guys to have a look at that as well. Now, innate behaviours that we've been discussing, like taxis, kineses, reflexes, these are basically behaviours, as we said before, which are designed to keep the organism in the optimal environment to ensure its survival. So, behaviours such as positive phototaxis and negative hydrokinesis result in organisms like maggots and woodlice being found where you find dark and moist conditions. So these environments are essential for the survival of these species because not only do they need to avoid desiccation, but they also need to avoid predators as well. So simple innate behaviours can often be viewed as reflexes to stimuli, but the analysis and interpretation of more complex innate behaviours can't be easily explained in terms of single reflex actions. And so for this reason, typically we study very simple organisms like these, where the innate behaviours are largely due to very simple reflexes. So let's have a look at reflex actions in a bit more detail here. So the events from the stimulus to the response in a reflex action are firstly that the stimulus occurs, which is a change in the internal or the external environment. This is followed by the detection of that change by an organ called a receptor, which is a sensory organ, and this, when it's stimulated, will generate an impulse sent along, or cause an impulse to be sent along a sensory or afferent neuron. Now this impulse, this signal, is then passed on to a coordinator or a coordination center, and usually this is in the central nervous system, so in other words the brain and the spinal cord. Now here relay neurons are involved and send an impulse from the sensory neuron to a motor neuron. So once the motor neuron sends out its efferent impulse, this reaches an organ called an effector, which is typically a muscle or a gland, and it's that effector which then causes the response to the stimulus, and the response again is the reaction to the stimulus. So this is the general scheme for a reflex action. Now, a simple reflex action that many of you have either observed or been subject to yourself when you go to the doctor is the knee-jerk reflex. Now, in general, we can define a reflex action as a rapid automatic response to a stimulus. Now, simple reflexes are inborn fixed responses to specific stimuli. Now, these can involve either the brain or the spinal cord. Now, the knee-jerk reflex, or the patellar reflex, this is conducted by striking the patellar ligament just below the patella or the kneecap. Now, this reflex helps us to maintain our posture and balance without actually thinking about it. So the knee-jerk reflex is pretty familiar to most people. And it's actually very simple. It only involves just two neurons, so a direct connection between a sensory and a motor neuron. And this is actually quite unusual because there's no relay neuron involved. So the reflex only involves one synapse for this reason, and for this reason it's referred to as a monosynaptic reflex. So if a person was to lose balance and to start to fall, then the kick resulting from the patellar reflex should give them enough height to allow the person to regain their footing. And that's really what the patellar reflex, the function of the patellar reflex in the organism. Now again, this is a rather unusual reflex because it only involves two neurons and a single synapse, but typically reflexes will involve three neurons: a sensory or afferent neuron, a relay neuron or intermediate neuron, and a motor neuron or efferent neuron. So spinal reflexes so the reflex actions, again, as we said before, involve the receptor, the sensory neuron, the motor neuron, and the effector. These form a nervous pathway which is sometimes described as a reflex arc. Now, the reflexes that operate via the spinal cord are referred to as spinal reflexes. Now, these include pathways that determine actions such as withdrawal responses. So you see here that this person has just injured their thumb by hammering on it. And so things like rapidly moving your hand away from some kind of painful stimulus, these are under the control of spinal reflexes. So reflexes such as the withdrawal response have obvious adaptive and survival value in that they protect the body from noxious or harmful stimuli. So the withdrawal reflex is an example of a more common spinal reflex involving three neurons. So if we take a transverse section through the spinal cord, the three neuron reflex is illustrated here by these neurons shown diagrammatically here. So reflex arcs will produce a very fast response because the impulses pass along a very simple nerve pathway involving very few neurons and synapses and no conscious thought or conscious control from the higher centers of the brain. So you start with a harmful stimulus. So the pain receptors in that part of the body are then stimulated. Those receptors will then cause a generator potential, which in turn causes a sensory neuron to fire an action potential, and that action potential, that impulse, will then move from that region of the body towards the spinal cord. The sensory neuron will then synapse onto an intermediate or relay neuron. The relay neuron then passes that signal along to the cell body of a motor neuron, and the motor neuron in turn will send the efferent impulse, the action potential, to the effector. In this case it would be the muscles of the arm, which would then cause you to withdraw your hand away from that noxious or harmful stimulus. Now, it is true that to some extent you can modulate your innate withdrawal responses. So even though the actual reflex arc only involves 3 neurons from the receptor to the effector, the brain can exert some influence. So reflex arcs can be modulated or modified to various synaptic connections. Not all of them, but some of them can be modified. Now these occur in the reflex arc between the relay neurons, sensory neurons and motor neurons, where there are ascending and descending tracts of neurons passing to and from the brain, which can influence the conduction of signals along this reflex arc. So for example, if you are carrying a hot object, or if you inadvertently pick up a hot object, typically your response would be the withdrawal response. The reflex arc would cause you to suddenly withdraw your hands away from the object and drop the object to prevent your hands from being burned. However, if you're picking up an object which is incredibly valuable to you, then to some extent you may well hang on to that object for longer in contravention of that innate reflex response. So now, the explanation as to how this works is somewhat beyond the level of advanced level biology, but I would encourage you guys to look this up because it is actually quite interesting. You might want to consider how this arrangement could modify the behavior or the response resulting from inadvertently handing a very valuable hot object when compared to inadvertently picking up a worthless hot object. So we've talked about we've mentioned so far that there are organs which are sensitive to changes in the internal and external environments, which are referred to as receptors. Now we need to get into some of this in a bit more depth. Now we're going to be exploring a couple of different types of receptors. We'll start with the Pacinian corpuscle, and then we'll move on to the rod and cone receptors in the retina of the eye. But again, we actually explore a lot of other different kinds of receptors in that course module on my website called Control Systems. But obviously I'm just going to stick to the ones that are explicitly mentioned in the advanced level biology specification from AQA in this webinar. So if you'd like more information on other kinds of receptors, by all means have a look at that. So let's start with the Pacinian corpuscle. Now there are many different kinds of receptors within the animal kingdom. Now each receptor is structurally and functionally adapted for the reception of a particular type of stimulus. Now, sensory receptors can either be the peripheral ends of sensory neurons, or they can actually be separate specialized cells which are adjacent and connected to those sensory neurons. Now, in either case, receptors will respond to changes in both the external and the internal environments, and as such, they're classified as either exteroceptors or enteroceptors. Now there is another class of receptors called proprioceptors, which are specialized receptors which respond to tension within tendons and muscles. Now proprioceptors are the receptors which allow you to be aware of the positions of your limbs and your joints in space, even when you can't see them. And so they're very important, among other things, in controlling and coordinating your movements. Now the important thing to bear in mind here is that receptors are transducers. They transduce the external energy of the stimulus into the code of nervous impulses. Now the application of the stimulus to a receptor leads to the formation of a generator potential, and the magnitude of that generator potential is determined by the stimulus intensity. So again, here we have a number of different kinds of receptors here. And we will be exploring some of these. We'll look at the Pacinian corpuscle, we'll also look at baroreceptors when we look at the control of heart rate, and we'll look at the photoreceptors found in the retina of the eye. But in general, sensory receptors monitor changes in either the external or internal environments, and the stimulus will generate the formation of a generator potential across the receptor cell membrane. Now the function of that generator potential is to trigger an action potential in a sensory neuron. So let's have a look at the Pacinian corpuscle, but again the functional anatomy and physiology of many different kinds of receptors are explored in depth in that Control Systems course module. So if you are interested in that, by all means check that out as well. So these are mechanoreceptors which are found in the skin and also in certain mucous membranes. Now these are responsive to pressure or any kind of mechanical stimulus which causes the deformation of the shape of that receptor. So the Pacinian corpuscle consists of a single sensory nerve ending, which is surrounded by a multi-layered capsule composed of connective tissue, with this gel-like fluid between those layers. So the nerve fibre is myelinated except for this terminal region, which is within the central core of the Pacinian corpuscle. Now it's important to remember here that there are no voltage-gated channels found in the nerve terminal, which is sensitive to the mechanical compression. The voltage-gated channels are, however, found at the nodes of Ranvier in the myelinated region of the corpuscle's nerve fibre. So here we see a micrograph of the Pacinian corpuscles in the skin, and you can see here that they're near the surface of the skin and they're surrounded by adipose tissue in the slightly lower layers underneath the cornified and dermal layers here. So, as we said before, the Pacinian corpuscle consists of a single sensory nerve fibre, the terminal region of which is enclosed in a multi-layered connective tissue capsule. The nerve fibre is myelinated, again, except for this terminal region, which is within the central core of the Pacinian corpuscle. And you see here a micrograph so that you can see the structures as they actually appear. So you have these concentric lamellae of connective tissue, and there's a viscous gel situated between these layers. So here we can see the non-myelinated terminal of the sensory nerve fibre from within the central core. This is a longitudinal section of the Pacinian corpuscle. What you're seeing here is a transverse section. So you're seeing the kind of transverse section through the non-myelinated tip here. Here's another micrograph to show you the structure here. Now here in the center you find stretch-mediated sodium channels within the non-myelinated terminal. Now these will open in response to pressure being applied to the Pacinian corpuscle, which is deforming the shape of the receptor. So the influx of sodium ions, which happens as a result of the opening of these sodium ion channels in response to pressure or shear on the Pacinian corpuscle, this results in a change in the membrane potential of that nerve fibre called the generator potential. Now the higher the higher the pressure or the mechanical deformation of the Pacinian corpuscle, the greater the magnitude of the generator potential. So we say that the generator potential is graded and, unlike the action potential, will vary with the stimulus intensity. So a generator potential is defined as a graded change in the electrical properties of the receptor cell membrane. So the generator potential, like an action potential, is a temporary change in the membrane potential. However, unlike the action potential, it will vary with the strength and the duration of the stimulus. So in other words, unlike the action potential, it does not display that all or nothing characteristic which you find with action potentials. Now, most generator potentials are created when a stimulus triggers the opening of sodium ion channels, which result in the depolarization of the cell membrane. Now, one exception to this is the effect of light stimuli on photoreceptor cells of the retina. Now, in that case, the stimulus triggers the closing of sodium ion channels, and the generator potential develops as the receptor cells become more negative or hyperpolarized. So here we see what happens when we compress a Pacinian corpuscle. So an applied pressure causes the deformation of the connective tissue capsule. This causes a stretching of the cell surface membrane of the non-myelinated portion of the sensory neuron, and that in turn causes the stretch-mediated sodium ion channels in the surface of that membrane to open, and this causes an influx of sodium ions to diffuse into the neuron. Now this causes a change in the membrane potential of the neuron, and this is referred to as the generator potential. So we can measure this by inserting electrodes into, or microelectrodes into the nerve fibre within a Pacinian corpuscle, and then monitoring the potential difference across that membrane as a pressure or stimulus is applied. So we see that this generates a generator potential, and then local currents will then travel from the stimulated unmyelinated tip of this nerve fibre towards the myelinated sections, with the strength of the current being dependent upon the magnitude of the generator potential. Now, if the generator potential reaches a critical threshold value, it will then trigger local currents of sufficient strength to stimulate the voltage-gated sodium channels at the first node of the sensory fiber, this first node of Ranvier. Now, when that happens, if we then insert a microelectrode into the myelinated region of the sensory fiber, we observe a typical action potential being recorded, which we can see using a cathode ray oscilloscope. So, if we imagine that we insert two microelectrodes here, X and Y, one into the non-myelinated tip of the nerve fiber in the Pacinian corpuscle, the second into the myelinated section here, and then record the voltage changes that occur across those membranes under conditions where we apply a light pressure, a medium pressure, and a heavy pressure, the recording from X is going to be measuring the generator potential. So we can see here clearly that as the pressure increases, the generator potential magnitude increases. But because microelectrode Y is being inserted into the myelinated section here, we see that in this section you either get a full action potential or you don't. Now, the action potential itself does not change in magnitude in response to the pressure. Instead, it will change in frequency. So action potentials will record the intensity of a stimulus not in terms of their magnitude, but in terms of their frequency, as you can see here. So pressure deforms the connective tissue capsule. The stretch-mediated receptors, stretch-mediated sodium ion channels in the non-myelinated section will then open, and this causes an influx of sodium ions into that nerve terminal, which then depolarizes the membrane. The higher the pressure, the greater the generator potential caused by that depolarization, until eventually it reaches a threshold value which triggers an action potential in the myelinated region of the nerve fiber. And then any increase in the generator potential simply causes an increase in the frequency, not the magnitude of the action potential. So the intensity of the stimulus is directly related to the frequency of impulses which are generated as action potentials in the myelinated region. And again, that is recorded by microelectrode Y here. So light pressure, as we can see here, gives rise to a sub-threshold generator potential which fails to trigger a full action potential at the first node of the sensory fiber. Increasing the pressure has given rise to a generator potential that has reached the threshold value, because here we see that we are getting action potentials, but then the higher the pressure beyond that, the greater the generator potential. We don't get any greater magnitude of action potentials, we simply get an increasing frequency here. So the Pacinian corpuscle typically generates the action potential at the beginning of a mechanical compression and another when the compression is then terminated. So we get an action potential in the myelinated region once the pressure reaches a critical value, but then we also get another action potential when the pressure is removed. So under conditions of no compression, there's no deformation, no generator potential, no action potential. Under conditions of compression, we get deformation, we get a generator potential which is over the threshold value, and that triggers action potentials. So during compression, what's happening is the viscous gel between those concentric lamellae of connective tissue is moving, and this allows the nerve terminal to resume its normal shape after the pressure has been removed. So under conditions of compression, we get deformation of the nerve ending. So as the pressure is removed, the whole corpuscle will then resume its original shape, with the nerve terminal becoming deformed again in response to that. So as the viscous gel flows back to its original position between the lamellae, the corpuscle will then return to its resting fully uncompressed state. So I see that we've reached the end of the first hour of this webinar, so I'm going to give you guys a 5-minute break here. After the break, we're going to go on to look at the receptors present in the retina of the mammalian eye. Okay, I see that quite a few questions have been posed in the private Zoom room here, so again, thank you for putting those in the Q&A box, guys. I'm going to address those at the end of this lesson. So let's get into the retina now. So So the human eye is a sensory organ, and it's located, and you find located within it the photoreceptors. Now the photoreceptors that we're going to be talking about, again, like all receptors, these are transducers. They convert the external energy of the light stimuli into the code of nervous impulses, which then go into the nervous system. So the photoreceptors in this case are the rod and cones of the retina. Now the eye is also structurally and functionally adapted for controlling the amount of light entering the eye and for focusing the incoming light rays into the retina. So let's have a quick look at the structure of the eye and remind ourselves of the various anatomical parts. So here we have the kind of glassy transparent layer which is called the cornea. Behind the cornea we have the gap through which light enters the eye, called the pupil. We also have the tough outer connective tissue covering of the eye, the white section, called the sclera. And we have the iris of the eye, which is the coloured portion around the pupil. So again, the structure of the eye, we have the sclera, which is the tough white fibrous layer. This protects the eyeball and allows for the attachment of external muscles which control the movement of the eye. We also have a pigmented layer just underneath the sclera on the inner surface of the eye called the choroid. Now this absorbs light and prevents internal reflection of the light rays. Here we have the layer of tissue called the retina at the back of the eye. Now this is the layer of tissue which contains the photosensitive rod and cone cells. Now in the retina there's a region of the retina which only contains cone cells. Now this region is called the fovea, and we're going to be referring to that a little bit later on. Here we have the optic nerve. And the optic nerve is the nerve responsible for transmitting the impulses to the brain, which are then interpreted in the occipital cortex, which is the region of the brain cortex at the back of the eye here, the back of the brain rather. Now, again, in the retina, there's a region of the retina referred to as the blind spot. Now, the blind spot is a portion of the retina which lacks both kinds of photoreceptors, both the rod and the cone cells. Now, there are two forms of liquid or jelly-like liquids in the eye. Now, in this compartment of the eye behind the lens you find the vitreous humor. Now this is a jelly-like fluid which fills the rear chamber of the eye. Now it's a jelly-like substance, and its function is both to transmit the light from the pupil through from the lens to the retina, but its other function is to maintain the shape of the eyeball. Now, down here we find the ciliary bodies, here and here. Now this alters the shape of the lens during focusing. Now these contain circular muscles which can contract and change the shape of the lens to focus light from distant or near objects onto the retina correctly. Here we have the iris. Now the iris will control the size of the pupil by either retracting or contracting. And obviously we have the pupil, which is a circular opening in the iris, and this directs light onto the lens. Here again we have the glassy layer of tissue called the cornea, and this refracts light and is also transparent. And around that we have a thin protective membrane called the conjunctiva, which protects the cornea. Now in this compartment at the front of the eye we have a different kind of liquid, which is a more watery liquid called the aqueous humor. Now this fills the front chamber of the eye in front of the lens, behind the cornea. And also we have these suspensory ligaments which attach the lens to the ciliary body, which contains those muscles which can either stretch the lens to focus light from distant objects or cause the lens to fatten to focus light from near objects. So this is the general kind of anatomy of the eye that we see here. And obviously we also have the lens, which is a gel-like structure which is responsible for focusing the image onto the retina correctly. Now the receptors that we're going to be talking about here are located again in this layer of tissue called the retina. Now this micrograph that you're seeing here is a section through the retina, and we can see here the rod and cone cells, and we can also see the bipolar neurons, which we're going to be looking at in a moment, which are responsible for conducting the impulses from the rod and cone cells to the optic nerve. Here we have the ganglion cells, again, which we're going to be referring to in a moment. So we need to understand how the rod and cone cells operate, but we also need to understand how the signals produced by these rod and cone cells in response to the incoming light are then transferred ultimately to the optic nerve. So in this diagram, the direction that the light is coming in is from this direction. So here we find the sclera. This is the choroid epithelium, which again is a pigmented layer of epithelium which is responsible for absorbing the light which is not intercepted by the photoreceptor cells that we see here, the rod cells and the cone cells. Now the rod cells contain a pigment called rhodopsin, and rhodopsin is only sensitive to low light intensities and is also unable to distinguish colours. Now in contrast, the cone cells contain a different pigment, iodopsin, and this is sensitive only to high light intensities and allows for the discrimination of colour. So down here we see a number of relay neurons. So these neurons here that connect to the cone cells, these are referred to as bipolar neurons. These transmit impulses from the photoreceptor cells to the ganglion cells. So these are the ganglion cells here, which are synapsing onto the bipolar neurons here. And we see that we have different types of bipolar neurons which synapse between the rod cells and the ganglion cells and the cone cells and the ganglion cells. And these excuse me, these have implications for visual acuity and various other aspects of the way that the optical system functions, which we're going to look at in a moment. So these are actually the neurons here of the optic nerve that we're seeing here. So let's look at those photoreceptors in a bit more depth here. So remember that rod and cones are transducers. They transduce the energy of light into the code of nervous impulses. So the outer segment of the rod cells have this thin rod-shaped appearance, whereas the outer segments of the cone cells have this more conical shape. And this is what gives the two different types of photoreceptor cells their names. So within this outer segment, there are numerous flattened membrane-bound vesicles called lamellae, which contain the photosensitive pigment rhodopsin. Now, the iodopsin in the cones is actually contained in infoldings of the cell membrane. Now, the inner segments of both of these contain many mitochondria and also contain the cell nucleus and also the synaptic base. So let's have a look at transduction in rod cells. Now, rod cells are extremely sensitive to even low levels of light. And so these are the photoreceptors in your retina which allow you to see under dim light conditions. So rhodopsin, which is sometimes called visual purple, it's a pigment molecule composed of the protein scotopsin combined with cis-retinal, which is actually a derivative of vitamin A. Now in the dark, there's a steady release of neurotransmitter from the synaptic base of the rod. So when the light is absorbed by the rhodopsin, the cis-retinal is converted into its isomer, trans-retinal, and that molecule then becomes unstable and breaks down. So this reaction results in the creation of a generator potential in the rod cell and a reduction of neurotransmitter release from the synaptic base. So the generator potential created in the rod, this is actually the result of the closure of some of the sodium ion channels and an increase in the negativity within the rod cells. So if the reduction in the neurotransmitter reaches a threshold, then these events will activate the adjoining bipolar neuron, which in turn will activate the adjacent ganglion cell, which then produces a nerve impulse, which is transmitted to the brain via the optic nerve. So let's have a look at how that works in rod cells. Now in the previous diagram you saw that the bipolar neurons which connect the rod cells, the synaptic bases of the rod cells, to the ganglion cells actually connect multiple rod cells to a single ganglion cell. So here we have a group of rod cells, here we have a bipolar neuron which synapses onto multiple of the synaptic bases of those groups of rod cells, and we have a single ganglion cell which synapses onto the bipolar neuron at the other end. and obviously the ganglion cell will then conduct that impulse to the optic nerve. So again, the breakdown of rhodopsin creates a generator potential that results in the passage of an impulse from the bipolar neuron to the ganglion cell and then via that to the optic nerve. So the impulse then travels along the optic nerve to the brain. Now the rods will continue to function as long as rhodopsin is resynthesized, and this requires ATP generated by the mitochondria in the inner segments of the rod cells themselves. Now at high light intensities, and again this is typical daytime light intensities, the rhodopsin breaks down rapidly and it can only be resynthesized in the absence of light. So when the rhodopsin is bleached in bright light, the eye is described as being light adapted. Now the rods will then cease to function, and then the cones are then the cells which are responsible for daytime vision. So rhodopsin is resynthesized in a process called dark adaptation. Now in total darkness, it takes about 30 minutes for all of the rods to become maximally sensitive again. Now, as we said before, cones are much less sensitive to light than rods are, and only begin to operate under normal daytime light intensities. So cones are able to differentiate colour, whereas rods only provide us with monochromatic vision or black and white vision. So cones again contain the pigment iodopsin, and that's sensitive only to high light intensities, and again allows us to perceive colours. So rods display a high visual sensitivity but a low visual acuity. Now you need to be able to explain the difference in visual acuity and visual sensitivity between rods and cone cells. Now the high visual sensitivity describes the ability of the rods to function at low light intensities. Visual acuity, on the other hand, it's about the sharpness of the image which is produced. Now rods display poor visual acuity because of the anatomical arrangement of the rods and the bipolar neurons within the retina. So in contrast to the rods, the cones provide us with good visual acuity, in other words sharp images, but poor light sensitivity. In other words, they're only sensitive to high levels of illumination. So cones generate nerve impulses in a similar way to the rods. Now the iodopsin absorbs the light, it breaks down, and this creates the generator potential in the cone cell and a nerve impulse ultimately in the optic nerve. So the breakdown of iodopsin is actually a slower chemical reaction than the breakdown of rhodopsin. So let's compare the visual acuity and sensitivity of those two types of photoreceptors here. So we see here that cones allow the discrimination of colour, whereas rods are responsible for monochromatic vision. Cones allow for good visual acuity, whereas rods provide poor visual acuity, and cones are said to be poor in their sensitivity to light, whereas rods show a good sensitivity to light. Now, retinal convergence, summation and amplification, these are the features that explain these different characteristics of rod and cone cells. So we need to make reference to these three concepts in order to explain the differences in the discrimination of colour, the visual acuity and the sensitivity of these two different types of photoreceptors. Let me make myself a little bit smaller on the screen here so I don't obscure some of that text. Sorry guys, let me just adjust that, as I see some of you are asking me to do that. And let's make this a bit smaller. There we go. Okay, sorry about that. Okay, so here we see here we can explain these terms, and by means of that we can explain the differences in the acuity, the sensitivity and the color discrimination between these two different types of photoreceptors. So we see here that many rods will converge and synapse onto a single bipolar neuron here, which in turn will connect to a single ganglion cell. Now what this means is that the brain, which receives one signal here via this fibre in the optic nerve, the brain is unable to distinguish where a given ray of light has struck the retina, because that ray of light may have struck any one of this number of different rod cells which are converge onto a single bipolar neuron, which in turn synapses onto a single ganglion cell. Now this is the reason why the resolution produced by rods compared to cones is poor. Now, in contrast to that, cones do not show convergence. They display a 1-to-1 ratio with the ganglion cells because each cone will only synapse onto a single discrete bipolar neuron, which in turn will synapse onto a single discrete ganglion cell, which then sends its signal via a single fiber in the optic nerve. So when cones are stimulated, the brain is able to resolve the impulses generated when light rays strike two very close positions on the retina. So, retinal convergence, coupled with the sensitivity of the pigment rhodopsin, explains the high sensitivity to light displayed by the rods. Now, at low light intensities, the graded generator potentials created in the individual rod cells will sum together and amplify the incoming light signals. Now, it's this amplification so this amplification is sufficient to generate an action potential in the associated bipolar neuron, and obviously then that synapses onto the ganglion, which then fires an action potential via the optic nerve fiber. So we mentioned that the amount of rods and cones found at various positions on the retina might vary. So if we so on this axis we have the angular separation from the fovea in degrees. Here we have the fovea location at 0. So the reason why we have this on this axis is because the retina is a curved surface. So if we take the reference point of the fovea as being 0, then we can measure the angular separation of various parts of the retina from that point. So we see here that cones are concentrated on the fovea centralis, the yellow spot of the retina. And so we can see here that the amount of cones is highest at the fovea, but the rods are absent from this region. We see that the density of rods is 0 at this location. But a few degrees away from that to either side, we see that the density of rods rises to a very high value and then spreads over a large portion or a large area of the retina to either side. So light striking the fovea produces the sharpest color vision. Now we also mentioned the blind spot. So if this is the retina layer here, and this is the sclera, here we can see the rod receptors, and we can also see some cone receptors here. This is the pigmented choroid layer of choroidal epithelium, and these are the ganglion cells. And obviously the ganglion cells all will send their fibers along the optic nerve here. So this here is the blind spot. The blind spot is sometimes called the optic disc. Now this is the area of the retina where the fibers of the ganglion cells exit along the optic nerve. Now it's called the blind spot because the photoreceptors are absent in this area. So incoming light rays that strike the blind spot produce no image. So how do we explain colour vision? Well, there's about 6 million cones in the human retina, and these are the photoreceptors responsible for colour vision. There are 3 types of cone: red, green and blue, and each possesses a different form of photosensitive pigment. So these cones are responsible for all the high resolution vision, and the eyes move continually to ensure that light from objects being viewed falls on the fovea, which has the highest concentration of cones, and it's in this way that we're able to distinguish different colours in our visual field. So the next thing that we need to go through is the control of the heart rate. So let's get started with that. Now, I'm not going to go into the functional physiology and anatomy of the heart because this is something that's covered in a different webinar, and it's also covered in a different course module on my website. So again, the webinar that's going to be covering the transport and exchange of substances is we're going to get into the gross anatomy and functional physiology and anatomy of the heart in that webinar. That will be, let's have a look here. I believe that will be, yeah, so that will be on the 5th of June at 5 p.m. And so we will get into that on that webinar. So I'm not going to go into the gross kind of anatomy and structure of the heart in too much depth here, but because we are going to be mentioning various parts of the heart, we'll go into that briefly here. But again, if you want more detail on that, you can either go into this exchange and transport course module, where we get into the heart and mass transport systems in a lot more depth, including the functional anatomy and physiology. But again, we also in the control systems course module on the control of the heart rate, I do go into the anatomy and physiology of the heart in a bit more depth there as well. So we're not going to go into that in too much depth other than to mention some of the major parts here. So here we have the vena cava, here we have the semilunar valves, we have the right atrium here, the tricuspid valve between the right atrium and the right ventricle. We have the septum, which divides the left and the right portions of the heart here. We have the aorta, the arch of the aorta shown here. The pulmonary artery here, which splits into the left and right pulmonary arteries. We also have the pulmonary veins entering the left atrium here. We have the bicuspid valve between the left atrium and the left ventricle. And of course, the left ventricle has a thicker muscular wall composed of cardiac muscle than the right ventricle here. Now, in terms of cardiac muscle, it's worthwhile mentioning here that unlike skeletal muscle, cardiac muscle has a very different morphology. So cardiac muscle tissue consists of branched, faintly striated fibers, each of which contains a centrally placed nucleus. Now, if you contrast this to skeletal muscle, which is multinucleate and linear, cardiac muscle is very different. Now, you also have the thickenings of the plasma membrane between the individual muscle cells, forming what we call these intercalated discs as well. So the cardiac muscle tissue forms a network due to its branching structure. Now, because of this, when one muscle fiber is electrically excited, an impulse is quickly spread in all directions to neighboring muscle fibers, such that that impulse spreads in all directions when the cardiac muscle is stimulated. Now, obviously, this is a diagrammatic representation here, but this is a photomicrograph of cardiac muscle fibers. So you can clearly see here that they are uninucleate and they are branched, and you can even see these intercalated discs here as well. Here you can see the nuclei of the cells. So we need to talk about the origin of the heartbeat. So the mammalian heart is special in that the electrical stimulation necessary for the contraction of its muscles originates from within the heart muscle itself. So we say that the cardiac impulse is myogenic. It occurs and arises within the heart tissue itself. Now, within the heart, there's a network of specialized cardiac muscle cells designed for initiating each heartbeat and also for the rapid and coordinated spread of excitation across the heart. Now, this network of specialized cardiac muscle cells is known as the conduction system, and the conduction system is composed of separate parts, which we're going to be making reference to. So we have the sinoatrial node, also known as the SA node or the pacemaker. We have the atrioventricular node, also known as the AV node. We have the bundles of His, and we also have conduction fibers called Purkinje fibers. Now, as the stimulus for contraction of the heartbeat originates from within the cardiac muscle itself, the heartbeat is described as being myogenic. So, the origin of the heartbeat is within a specialized patch of cardiac muscle fibers here, which is referred to as the sinoatrial node. Now, this is actually located within the wall of the right atrium. So there's another node of specialized cardiac fibers known as the atrioventricular node or the AV node, and this is also located in the right portion of the septum between the two atria, and it's actually quite close to the atrioventricular valves here. So this is the AV node here. Now, from the AV node, you have these bundles of fibers which are responsible for conducting the impulse from the AV node down through the septum and along through here. So these are called the bundles of His, and these are located in the septum. Now, the bundles of His divide into the left and right bundle branches that we see here, and ultimately those bundle branches terminate in these branching fibers called Purkinje fibers, which are located in the walls of the two ventricles. So, when the sinoatrial node emits a spontaneous electrical impulse, these impulses will then spread rapidly outwards across the cardiac muscle fibers across the walls of the two atria. Now, as they spread, they will then cause a wave of contraction within the atrial walls, and this triggers the contraction of the atria, which is referred to as atrial systole. Now, when the electrical impulses then reach the border between the atria and the ventricles, they're then blocked by a band of non-conducting fibrous tissues. Now, in order to reach the ventricles, the electrical impulses first have to pass through the atrioventricular node. Now, this slows down the speed of electrical transmission. Now, this delay, called the atrioventricular delay, this is extremely important because it allows the atria to finish contracting before the ventricles begin to contract. So, once you reach the once the impulse reaches the atrioventricular node, the impulse is then conducted from the AV node along the bundles of His. The bundle fibers divide into numerous Purkinje fibers, which then permeate the ventricular cardiac muscle. And then the spread of electrical impulses through the ventricles is what triggers ventricular systole or ventricular contraction. So, to summarize, we can summarize what we've just been talking about in this diagram here. Now, the whole purpose of the conduction system of the heart that we're referring to is to ensure the correct order of contraction of the ventricles, of the atria, followed by the ventricles. And this in turn is what ensures the movement of blood within the chambers of the heart in the correct directions at the correct times. So, the contraction of the muscles of the heart is associated with electrical changes called depolarizations, and these changes can be detected by electrodes attached to the surfaces of the body. Now, when the electrical changes associated with the cardiac muscle contraction are then inscribed on a ruled strip of paper, they provide a diagram called an electrocardiogram. Now, this is a permanent record of cardiac activity and looks something like this. So, in order to understand the electrocardiogram or the ECG, it's necessary to consider the electrical properties of the cardiac muscle. So, during diastole, when the cardiac muscles are at rest, they display an unequal distribution of charge across their membranes. Now, at rest, the cardiac muscle cells are internally negative and externally positive. Now, this charge difference creates a small voltage across the cardiac muscle cell membranes. Now, this can be detected by electrodes placed across the chest wall. So, when the cardiac muscle cells are at rest and therefore internally negative and externally positive, they're described as being polarized. Now, when a stimulus from the sinoatrial node spreads along the cardiac muscle cell membranes, there's a reversal of that charge distribution, so that when the stimulus arrives, that charge distribution reverses, where the internal portion of the membrane becomes positive and the external side of the membrane becomes negative. So, under this condition, the cell is described as being depolarized. So, depolarization then stimulates the cardiac muscle cells to contract. So, the depolarization results in a voltage change across the membrane, and again, this can be detected by electrodes applied to the chest. Now, remember that cardiac muscle cells form interconnected networks, so the stimulus originating from the sinoatrial node spreads from cell to cell, creating a wave of depolarization throughout the muscle cell network. So, depolarization results in muscle contraction, and therefore a wave of contraction spreads across that network of muscle cells. So, as the stimulus dies away, the muscle cells then return to their polarized state and cease their contraction. They begin to relax. So, this event is termed repolarization. Now, the ECG electrodes will detect the waves of depolarization and repolarization occurring during the cardiac cycle, and so the ECG trace is actually a record of all of these events happening. So here is a typical ECG trace from a healthy heart. Now, we can break that down so that the ECG trace for each heartbeat displays different portions. So here we have 2 heartbeats and the ECG traces caused by those 2 heartbeats. Now, each of these is composed of different sections which we can label: a P wave, a QRS complex or QRS wave, and a T wave. So each beat of the heart produces these waves in sequence. Now, in order to interpret these, we need to understand what is causing each one of these waves. Now, the P wave is the result of depolarization spreading from across the atria from the sinoatrial node, and it coincides with atrial contraction or atrial systole. The QRS wave, or QRS complex, this is the result of the depolarization of the ventricles, and this coincides with ventricular systole. And finally, the T wave, this is the repolarization of the ventricles as they begin to relax. Now, the repolarization of the atria is not detected because the small voltage change involved in that repolarization of the atria is masked by the larger QRS wave. So, this is a typo here. Shouldn't say 9.3, sorry, this should say bracket 3. So, we can also talk about the intervals, the time delays between each of these waves. The PR interval is the time which elapses between the events of atrial systole, which are responsible for the P wave, and the beginning of ventricular contraction, which is responsible for the QRS complex. So this period represents the time taken for the impulse to spread from the sinoatrial node through the atria, plus the delay in transmission from the to the atrioventricular node, together with the conduction time through the bundles of His and the Purkinje fibers. Now we can also talk about a TP interval, the interval between the T wave of one heartbeat and the P wave of the next heartbeat. So this is the time spent by the heart in relaxation or diastole before the next atrial systole begins. So using our knowledge of the ECG and the actual electrical events happening in the heart which are responsible for each one of those P, QRS and T waves, and also the intervals between them, a cardiologist can actually diagnose various pathologies, various things going wrong within the heart tissue, within the valves and so on, when those different waveforms change. So simply by looking at an electrocardiogram, a cardiologist can interpret changes in the T wave, changes in the in the intervals, changes in the QRS complex, changes in the P wave. All of those can be interpreted by a cardiologist to diagnose underlying pathologies of the heart tissue. Now, I know many of you present in the private Zoom room here are aiming to get into medical courses at university. Those of you who do, you're going to spend a lot of time looking at different ECGs from patients with various medical conditions, so that you yourselves will be able to identify those pathologies from those ECG traces. So let's look at the control of the heartbeat then. So although the origin and the transmission of the heartbeat are properties of the heart itself, it is necessary for the heart rate to be modified to meet the demands of the body as the demands of the body changes. Now, the heart rate is regulated by both the nervous and the hormonal systems of the body. So the autonomic nervous system is responsible for the regulation of the heart rate. The autonomic nervous system has two divisions, the parasympathetic and the sympathetic nervous systems. So let's look at the influences of both of those. So the autonomic nervous system is, generally speaking, the nervous system which controls the aspects of your body's function which are not under conscious control. And it's divided into two branches, the sympathetic and the parasympathetic. Now, in the context of the control of the heart rate, it's the sympathetic nerves which release the neurotransmitter noradrenaline from their terminals, whereas the parasympathetic nerves release the neurotransmitter acetylcholine at their terminals. Now, the heart is actually supplied with both sympathetic and parasympathetic nerves, and the chemicals that they secrete onto the cardiac muscle tissue then modifies the heart rate. So, two autonomic nerves link the cardiovascular centers in the brain with the sinoatrial node of the heart. So you have a sympathetic nerve which, when stimulated, will release noradrenaline at its terminus onto the sinoatrial node. Now, the effect of this chemical is that it causes the sinoatrial node to increase the rate of pulses that it generates, and this in turn will speed up the heart rate. Now, you also have a parasympathetic nerve which is a branch of the cranial nerve called the vagus nerve. Now this, when it's stimulated, releases acetylcholine at its terminus onto the sinoatrial node, and this has the opposite effect. It slows down the heart rate generated by the sinoatrial node. Now, there are also numerous sympathetic nerves which also innervate or link to the walls of the two ventricles, and these cause an increase in the force of contraction. So the heart rate is therefore determined by the balance between the sympathetic and the parasympathetic activity which innervate the sinoatrial node of the heart. So sympathetic activity dominates during periods of stress and exercise and excitement, whereas parasympathetic activity dominates during periods of rest, relaxation and sleep. So increased sympathetic activity also stimulates the release of the hormone adrenaline from the adrenal glands, and the adrenaline in the bloodstream then causes an increase in both the heart rate and the force of contraction. So here we see the medulla of the brain, and it's in this location here that we find the cardiac center, which is responsible for the sympathetic and the parasympathetic innervation, which then causes changes in the heart rate and modifies the heart action. Now, changes in blood pressure can also control the rate at which the heart beats through a negative feedback mechanism. So let's have a look at how this works. So again, remember that the origin of the heartbeat is myogenic, but the rate of the heartbeat can be varied by both nervous and hormonal mechanisms. So we have here a transverse section through the medulla of the brain, where you find two centers. You find a cardio-acceleratory center and a cardio-inhibitory center. Now, both of these receive incoming information via sensory afferent neurons coming from baroreceptors located in the carotid sinuses, which are located on either side of the neck, where the carotid artery branches on either side. But you also have baroreceptors located in the arch of the aorta as well. Now, the word baroreceptor refers to receptors which are responsive to pressure. So these receptors located in these blood vessels are responsive to the pressure of the blood in those blood vessels. And the way that they detect that is that they detect stretch in the walls of those blood vessels. So the baroreceptors detect and respond to changes in blood pressure, and they transmit impulses to the cardiac centers within the medulla, and those impulses carry information about the level of pressure in those vessels. So baroreceptors will transmit impulses to the cardio-acceleratory center when they detect a fall in blood pressure. Now, in response to that, the stimulation of the cardio-acceleratory center activates the sympathetic innervation of the heart, and these fibers pass from this center down the descending tracts of the spinal cord along the spinal nerves to the heart, and these sympathetic fibers will innervate both the sinoatrial node and the atrioventricular node, as well as the muscular walls of the ventricles. And the effect of that innervation is that the nerve terminals of these sympathetic neurons release noradrenaline onto the heart cells, and this causes an increase in the heart rate and also an increase in the force of contraction of the heart. So again, this is a negative feedback mechanism, because what happened was the pressure dropped, and so in response to that, once the cardio-acceleratory center detected that via the afferent impulses traveling from the baroreceptors, it then caused the heart to increase its rate and increase its force of contraction, and of course that will have the effect of increasing the pressure. Now, in contrast to that, baroreceptors will transmit impulses to the cardio-inhibitory center when they detect a rise in blood pressure. Now, in response to that, the stimulation of the cardio-inhibitory center activates the parasympathetic innervation of the heart. Now, these parasympathetic fibers pass from this center in the medulla down the descending tracts of the spinal cord along the spinal nerves to the heart, and there they will release acetylcholine onto the heart cells from their nerve terminals, and this in turn will cause a decrease in the heart rate. So we've mentioned quite a lot about nerve cells, nerve fibers, and various parts of the nervous system. So let's have a look at the nervous system, and in particular how neurons will generate action potentials and impulses. So neurons are the nerve cells which are specialized for the transmission of electrical or electrochemical impulses to other neurons, muscle cells and gland cells. Now within the mammalian nervous system there are three functional types of neurons that we referred to before. There are sensory or afferent neurons that transmit impulses from receptors towards the central nervous system. There are motor or efferent neurons, and these transmit impulses away from the central nervous system to the effectors. And there are relay neurons, which transmit impulses between sensory and motor neurons, and these are located within the central nervous system, within the brain and the spinal cord itself. Now be careful, because there are two different spellings that can be used for this word. Now this is the British spelling with an E between the N and the S on the end, and this is the American spelling. Now the spelling neurone, the British spelling, is used through these presentations here. So this diagram here shows the overall structure of a single motor neuron here. We see that we have the cell body containing a nucleus. We have the cell surface membrane, and coming from the cell body we have a long projection which is called the axon, and the axon in this neuron has these myelin sheaths around it in these sections. Between the myelin sheaths we have these nodes of Ranvier which are unmyelinated. Now the myelin sheath itself is produced by an accessory cell called a Schwann cell, and it's basically a concentric wrapping of that Schwann cell around the axon. Now within the axon you find protein microtubules and microfilaments. And from the cell body there are also these tree-like projections called dendrites, the singular being dendron, and it's through the dendrites that these cell bodies can synapse directly onto other cell bodies. Now you can also have the synapse between one cell and another via synaptic connections between the axons of surrounding cells, which are coloured darker blue here. So this would be an axodendritic synapse, because it's a synapse between the axon of one cell and the dendron of another cell. Now at these synapses, and again we're not going to go into synaptic structure in this particular revision webinar, because I've already uploaded one of the free videos from my website covering synaptic transmission, but in general synapses have this structure where we have the presynaptic neuron with its synaptic knob here containing vesicles which have the neurotransmitter in them, and on the postsynaptic membrane there are cell surface receptors which are specific for those neurotransmitters. So let's have a look at the structure of a motor neuron. So here we see the dendrites. Now these have the effect of increasing the cell surface area of the cell body. Here's the cell body surrounding the nucleus. This portion here is called the axon hillock. Here is the axon containing the axoplasm. Here is the myelin sheath, which again is made by the Schwann cells. These are the non-myelinated axon terminals, and you have the synaptic knobs, which can either synapse onto other neurons, or they can also synapse onto muscle cells, in which case you'd refer to them as neuromuscular junctions. So in this case the effector is a muscle cell group. So the direction of the impulse travels from the cell body down the axon to the synaptic knobs. So motor neurons carry nerve impulses from the central nervous system, in particular the motor cortex, but also from the spinal cord in the case of motor neurons involved in reflex arcs. And they carry those impulses to effector organs, which can either be muscles or glands or other cells. Now in contrast, sensory neurons have a very different structure. These convey impulses from sense organs into the central nervous system. So the CNS processes the impulses, and this gives us our perception of our senses such as sight, smell, touch, etc. So again, here we have nodes of Ranvier on the myelinated section of this nerve cell. Here we have the dendrites, and again, in contrast to the motor neuron here, the cell body is actually located in the middle of the axon here. So the cell body surrounds the nucleus. And here we see the axon containing the axoplasm and the myelin sheaths of the Schwann cells. Now at the other end, we have the sensory nerve ending. Now some sensory neurons have their own sensory nerve endings, which are transducers for various stimuli. But as we said before, sometimes you will have sensory neurons which will synapse onto specialized receptor cells. Now in this case, the direction of the impulse is from the sensory nerve ending to the central nervous system. Now, nerves are macroscopic structures which are composed of bundles of multiple nerve fibers or nerve cells. So nerves are bundles of nerve fibers, and each nerve fiber is the long extension of a single neuron. So in other words, the axon of a motor neuron, for example. So here we have bundles of nerve fibers. These are myelinated nerve fibers in those bundles, and these bundles are surrounded by connective tissue. So the nerves may contain entirely motor fibers or entirely sensory fibers or a mixture of both. So here we see a transverse section of a whole human nerve, and we can see here the myelinated neurons within that nerve. So resting neurons, like all other human cells, are positive on the outside and relative to the inside as a consequence of the unequal distribution of charged organic molecules across the membrane. Now the opposite charges are insulated by the high lipid content of the plasma membrane, such that a voltage or potential difference called the membrane potential exists across that membrane. Now cells that exhibit a resting membrane potential are said to be polarized. So the resting potential, or the electrical difference across a neuron cell membrane when the neuron is inactive, is usually equal to about minus 70 millivolts for most neurons. Now, the presence of an appropriate stimulus at a point on the neuron cell membrane will trigger an action potential at the point of that stimulation, and this is shown by this graph here. Now, an action potential is a temporary reversal of the resting membrane potential. So in other words, the inside of the neuron membrane becomes positive relative to the outside. So we see here that we go from the resting negative membrane potential. During the action potential, this reverses to a positive membrane potential. So this temporary reversal of charge across the membrane of a resting neuron, this is described as a depolarization. So this portion of the action potential is referred to as depolarization. Now, the restoration of the resting membrane potential, this is described as a repolarization, and there's a slight overshoot of the membrane potential to a value that is more negative than the resting membrane potential. This is described as a hyperpolarization until the membrane potential returns to its resting membrane potential. So there's quite a few different kinds of charged particles in the fluids, both inside and outside of neurons. So sodium ions and potassium ions are the ones which are present in the highest concentrations, and it's the distribution of these ions across the neuronal cell membrane which exerts the greatest effect on the membrane potential. So before we get into that and start talking about ion channels, I see that we've come to the end of the second hour of this webinar. So again, I'm going to give you guys a 5-minute break. After the break, we're going to return and talk about how these ions and the movements of these ions across the neuronal cell membranes are responsible for both the resting membrane potential and also the action potential. So I'll come back after the 5-minute break. Okay, so the passage of charged particles across the lipid bilayer, this is what's responsible largely responsible for the maintenance and of the resting membrane potential and also the establishment of an action potential. So this is achieved only when there are specific protein channels which allow these ions to move across the membrane, and also carrier proteins which are present. So this is a kind of diagram which shows in a very simplistic way how that works. So we see here an axon. We see that there are large negatively charged protein molecules on the inside in the axoplasm. Excuse me. And you also have these ion channels. Now, some of these ion channels are ungated, so in other words, these will allow the movement of ions down their concentration gradients, but there are also channels which are gated, as we'll see. So there are two types of ion channels in the neuronal membrane. There are voltage-gated channels, which open and close in response to changes in the voltage or the membrane potential across the membrane. Now, these channels are closed when the neuron is at rest. But you also have so-called leak channels. These are protein channels which remain permanently open. So there are leak channels for potassium ions, but there are no leak channels for sodium ions. So the channels which allow potassium ions through are denoted in blue here. So this would be a voltage-gated potassium channel. This would be a leak potassium channel. But you see here that the only types of sodium channels which are present are voltage-gated sodium channels. There are no leak sodium channels present. Now, in addition to that, you also have the sodium-potassium ATPase pump, which constantly pumps sodium out of the cell and potassium into the cell by making use of the hydrolysis of ATP. So, let's see here. Alrighty. So we see that there are voltage-gated channels for both potassium and sodium ions, but there are only leak channels for potassium ions present. Now that's going to be very important for the explanation of the generation and the maintenance of the resting membrane potential as well as the action potential. So let's look at the resting membrane potential first. So the potential difference across the unstimulated neuron membrane, so in other words the resting membrane potential, this is actually largely due to the activity of the many sodium-potassium pumps within the membrane, together with the existence of these numerous leak channels for potassium ions. So the permanently operating sodium-potassium pumps will maintain a chemical concentration gradients for both sodium and potassium ions by actively transporting these ions against their concentration gradients. So there's the constant movement of sodium ions out of the axoplasm, and there's the constant movement of potassium ions into the axoplasm. Now, the ATP-driven sodium-potassium pumps transport the sodium ions out of the axoplasm, as we said before, and the potassium ions into the axoplasm. So this creates a high internal potassium ion concentration and a high external sodium ion concentration. Now, it's the operation of these pumps, which are under constant operation, and it's this which creates this steep concentration gradients for both of these ions. But remember that there are these leak channels present in the membrane only for the potassium ions. Now, the permeability of the neuron membrane to potassium ions is therefore significantly greater than the permeability of the membrane for sodium ions. So what this means is that the potassium ions begin to diffuse out of the axoplasm down their concentration gradients through these leak channels. So now we have this situation here. So within the axoplasm of the neuron, there's a number of negatively charged ions, and these are largely just proteins. Now, the proteins are too large to pass through the membrane. So as the positively charged potassium ions leak out of the neuron and very few sodium ions leak in, this means that the inside of the neuronal membrane becomes increasingly electrically negative relative to the outside of the neuronal membrane. So the development of an internal negative charge slows down the outward movement of potassium ions through the leak channels. Now, this electrostatic force of attraction between the negatively charged protein ions, this attracts the positively charged potassium ions into the neuron. So as the membrane potential reaches approximately -70 millivolts, the electrical force attracting the positive sodium ions into the neuron is equal to the chemical concentration force driving the potassium ions out of the neuron. So we at the resting membrane potential at -70 millivolts, these two opposing forces are equal in magnitude. So at the equilibrium potential of -70 millivolts, we can say that the electrochemical gradient for potassium ions disappears and there is no net movement of potassium ions. So as many potassium ions enter the neuron as are leaving the neuron. Remember, there is no net movement, but that does not mean there is no movement of potassium ions. It's just that the movement in due to or the movement out due to their chemical concentration gradient is exactly balanced by the electrical gradient, which tends to attract the potassium ions back into the cell. So the resting membrane potential is therefore maintained at a steady -70 millivolts, and the membrane is now described as being polarized, in other words, internally negative and externally positive. So this explains the generation of the resting membrane potential, but how do we explain the generation of the action potential? So an action potential, remember, is a membrane potential that temporarily reverses in response to a stimulus and then returns to its original resting membrane potential. Now, it's changes in the permeability of the axon membrane to sodium and potassium ions, which are responsible for the depolarization and the repolarization phases of the action potential. So, changes in the permeability of the axon membrane during the action potential are due to the opening and closing of those voltage-gated ion channels which control the movement of sodium and potassium ions through the membrane. So, the unstimulated neuron is polarized, as we said before, and the resting membrane potential is about -70 millivolts. Now, at this membrane voltage, the voltage-gated sodium channels that you see here are in a closed position, as you see here. So here, on this side of the membrane, we have the extracellular fluid, on this side we have the axoplasm, and under conditions where we're at the resting membrane potential, the inside is negative with respect to the outside to a potential difference of approximately -70 millivolts. Now at this voltage, the voltage-gated channels, remember voltage-gated means essentially voltage-controlled. At this membrane potential, these voltage-gated sodium channels are in the closed position. Now, when an area of the neuronal membrane is stimulated, so for example by an incoming action potential from a neighboring neuron which synapses onto that region of the membrane, depolarization begins, and the shift in the membrane potential to around -55 millivolts, this triggers the opening of a few of these voltage-gated sodium channels, and this in turn causes sodium ions to rapidly enter the axoplasm down their steep concentration gradient. And again, remember, that concentration gradient was established by the constant action of those ATP-powered sodium-potassium pumps. Now, the entry of the sodium ions into the axoplasm continues until the membrane potential reaches a value of about plus 40 millivolts. Now, this is the maximum value that we saw before of the action potential. Now, at this peak value of plus 40 millivolts, the voltage-gated sodium channels become inactivated and block any further entry of sodium ions into the axoplasm. So now we have an inactivated sodium channel. Now, it's at this point that the voltage-gated potassium channels now start to open relatively slowly, allowing potassium ions to move outward across the neuronal membrane and re-establish the resting membrane potential during that process, that phase of the action potential that we call repolarization. So now, with the inactivated sodium channels, the voltage-gated potassium channels open, and this allows an efflux, a movement outwards of potassium ions down their concentration gradient, and this re-establishes the resting membrane potential. So during this period, the membrane is unresponsive to any further stimuli while the voltage-gated sodium channels are inactivated. Now, this refractory period, as it's called, lasts until the relatively slow outward flow of potassium ions returns the membrane back to its resting value of -70 millivolts. So the voltage-gated sodium channels return to a closed rather than inactivated position once the resting membrane potential has been restored, and this area of the membrane can now respond to further stimulation. So that explains the generation of the action potential and the restoration of the membrane back to its original resting membrane potential. So to summarize, at the resting membrane potential, the voltage-gated sodium channels are closed in the resting neuron. At this point, a stimulus on the neuronal membrane causes a local depolarization of the membrane, and this causes a slight depolarization to the threshold potential of minus 50 millivolts. So if the change in the membrane potential due to the stimulus reaches this threshold value, this causes a few of the voltage-gated sodium channels to start to open. But because of the steep concentration gradient, you get a rapid influx of sodium ions, and this leads to further depolarization and the opening of more and more voltage-gated sodium channels, and this is a process of positive feedback. Now, once the membrane potential reaches this peak value, the membrane potential causes the voltage-gated sodium channels not to close, but to become inactivated. Now, the voltage-gated potassium channels now start to open relatively slowly, and this allows potassium ions to move outwards directly across the neuronal membrane, and this causes the repolarization of the membrane. Now, you also get this hyperpolarization because the repolarization overshoots the resting membrane potential and causes the membrane to become even more negative for a short period of time. And this is called hyperpolarization. Now, it's at this point that the sodium-potassium pumps re-establish the resting potential following the hyperpolarization, and then you go back to the beginning where you've got the resting membrane potential. And it's at this point that the sodium channels return from the inactivated position back to the closed position, and now that membrane can be again depolarized by another stimulus to cause another action potential. So, to summarize, this again is an animation just summarizing what I've just said, and I can use this as an excuse to take a sip of my tea. So you need to be familiar with all of those events to be able to explain the establishment of the resting membrane potential as well as the action potential. So you also need to be aware of the events that take place which cause that action potential to be transmitted along the axon from the point where it's generated. So let's look at the transmission of an action potential first along an unmyelinated neuron, and then we'll look at what happens with a myelinated neuron. So all action potentials are propagated or spread as self-generating waves of depolarization along the length of a neuron. Now these self-propagating action potentials traveling along the axon are described as nerve impulses. So when an action potential occurs on a segment of an axon, it produces local currents which stimulate the adjacent areas of the neuronal membrane. Now this area of the membrane depolarizes, and this initiates another action potential ahead of the previous one. So as action potentials are spread or propagated along the axon, local currents produced spread out in all directions, but the nerve impulse only travels in one direction along the axon, so it's said to be unidirectional. Now the reason for the unidirectional nature of the nerve impulse is because the area of the axon membrane that's recently depolarized is undergoing repolarization, and the voltage-gated sodium channels are in the inactivated position. So the membrane is in what we call a refractory state, and in that state it's temporarily incapable of producing another action potential. Now again, this only applies to the area of the membrane which has just fired an action potential. That will stimulate the neighboring areas of the membrane accordingly to generate their own action potential, as we'll see. So inactivated sodium channels in previously unstimulated or previously stimulated areas of the membrane, this is what causes that area of the membrane to enter that refractory period. So this is the direction of propagation in relation to time along this axon. And so this is what happens here. And this is how the action potential is propagated as a wave of depolarization. Now we're talking here about an unmyelinated neuron. So when an action potential occurs on one segment of the non-myelinated axon, it produces local currents that act as the stimulus for the adjacent areas of the membrane, which in turn depolarizes and generates an action potential. So this process continues unidirectionally along the axon so that the impulse travels down the length of that axon in one direction as a self-propagated wave of depolarization. So again, the refractory period is the time during which the neuronal membrane is unresponsive to further stimuli. Now we can break this down into the absolute refractory period, and this coincides with the process of repolarization in a previously active region and lasts for 1 millisecond. Now during this period, the area of the neuron membrane that's just been activated is totally unresponsive to any further stimuli. Now we can also talk about the relative refractory period. Now this lasts for approximately 4 to 10 milliseconds, and it's a period following the absolute refractory period where the membrane will respond only to high intensity stimuli. So the excitability of the neuron membrane is below that of the resting neuron during these two refractory periods, both the absolute refractory period and the relative refractory period. So let's look at the excitability of the neuron before and after a neuronal impulse. So on the vertical axis of this graph, we have the neuron excitability in arbitrary units. So in other words, this is how amenable to generating an action potential the neuron membrane is following a stimulus in arbitrary units. The higher the excitability, the more likely that neuron is to fire an action potential at a given level of stimulus. So we see here that the resting excitability of the membrane is at this value here. Once the stimulus occurs, the excitability of that portion of the membrane drops down to 0 for the absolute refractory period, or during this absolute refractory period. Now, following the absolute refractory period, the excitability of that portion of the membrane slowly increases back to its resting state during the relative refractory period. So we see here that the absolute refractory period, during which the membrane is completely unresponsive to stimuli, is relatively short. It's about 1 millisecond. But then following that, you have a relative refractory period during which the excitability of the neuron is still low to begin with, but then increases back to its resting excitability. So together, those two refractory periods, the absolute and the relative, make up the total refractory period for the neuron. So a new stimulus applied to an area of the membrane that's just been active will not initiate another action potential during the absolute refractory period, during that 1 millisecond. But during the relative refractory period, a stimulus of high enough intensity may depolarize the region of the membrane that's just been active. So there are a few features of action potentials which distinguish them from things like generator potentials. So firstly, they are said to be all or nothing, and this is sometimes described as the all or nothing law. So this basically describes the fact that each neuron has a threshold of stimulation below which it will not fire an action potential. Now, above this threshold, all stimuli, regardless of strength, bring about an action potential whose magnitude is independent of the strength of the stimulus. Now, the other aspect of action potentials that we mentioned in passing earlier is the coding of nervous information. So as the action potential is always of a fixed amplitude or magnitude, then the code of the nerve impulse is a frequency code, so that the stronger the stimulus, the greater the frequency of the action potentials generated and the greater the amount of neurotransmitter released at the neuron terminals. The speed of the transmission of action potentials is affected by the properties of the neurons themselves. So for example, the larger the diameter of the axon, the faster the rate of conduction of that impulse. And the reason for this is due to the larger area of the membrane available for the ion movements and the sodium-potassium pumps and the lower resistance to local intracellular current flow. Now, the other thing to mention here is that myelinated neurons conduct impulses much faster, and the reason for this is due to the presence of the myelin sheath. So we've looked at the conduction of an action potential across an unmyelinated neuron. Let's now take a look at what happens when an action potential is conducted across a myelinated neuron. So in myelinated neurons, as we saw before, the axon membrane is insulated by a fatty myelin sheath, which is formed by the accessory cells which wrap themselves around the axon, called the Schwann cells. Now, this sheath insulates the axon against electrical interference from neighbouring cells, but this insulation is also incomplete because there are regularly spaced, uninsulated gaps between those adjacent myelin sheaths. So these gaps are called the nodes of Ranvier. Now, due to the insulating properties of the myelin sheath, the depolarization of the axon membrane can only occur at these nodes. So, when an action potential occurs at a node along an axon, it produces electrical currents which depolarize the next node along. So the action potential generated at this next node produces in turn electrical impulses and currents which depolarize the next adjacent node, and so on. So you still get this unidirectional propagation in relation to time. So this is how the action potential jumps essentially from one node to the next. So in myelinated neurons, the nerve impulses are propagated by what we call saltatory conduction. This type of jumping effect is called saltatory conduction. So the action potential appears to jump between node to node. Now, in myelinated neurons, the speed of transmission of action potentials is actually much greater than the speed observed in non-myelinated neurons of a similar diameter, and the reason for this is because of this saltatory conduction. Unlike the unmyelinated neuron, the action potential doesn't have to sweep across the entire length of the axon membrane. Instead, it jumps between these nodes, and in so doing, it takes less time to cover the same distance along the axon. Now, as we said before, the speed of transmission of action potentials along the neuron is also affected by the diameter of the axon, and this holds true as well for myelinated neurons. So just like unmyelinated neurons, the larger the diameter of the axon, the faster the conduction of nerve impulses along that neuron. So let's move on to the next section. As I say, I'm not going to go into synaptic transmission here, just because I have uploaded a free video for to cover that to both the front page of the website at invisible-college.com, but also to the YouTube channel that I said before. So if you're looking for details of that, then by all means have a look at those videos, and if you have any questions, then go ahead and type those questions either in the comments on the YouTube pages for those videos or on the lesson pages on my website as well. So let's get into homeostasis. So homeostasis is an essential condition for the maintenance of life, and it's defined as the maintenance of a constant internal environment despite fluctuations in both the body's activities and in the external environment. So various body systems will work cooperatively to ensure that, for example, the composition of blood and tissue fluid remains within tolerable limits. So these types of homeostatic controls ensure that the factors such as blood, pH, temperature, water potential are kept within normal limits. Now, cellular biochemical reactions are controlled by enzymes, and these enzymes will only operate at optimal levels when they are within very narrow ranges of these sorts of parameters like pH and temperature. So it's incredibly important that these parameters within your body compartments and tissues are kept within very narrow limits, and it's these processes of homeostasis which are responsible for that. So homeostasis is usually achieved by a process that we call negative feedback. So negative feedback mechanisms will always involve some kind of deviation of some measurable factor from its normal value or set point, receptors that detect that deviation from the norm, and effectors which are activated by the receptors and correct that deviation by mechanisms that restore that norm or set point. So we see here that deviation above or below from the norm will be detected by receptors in both cases. These then stimulate effectors, which then initiate corrective mechanisms which restore those parameters back to the norm. So again, this is shown by this chart here. Let me just bring that over here. So we start with some parameter which is at some value, which we call the norm or the set point. Now when that value deviates either below or above the norm, this is classified as a stimulus, which is detected by a receptor, which then triggers an effector, which then triggers a response, which then restores the norm. And the same thing happens when the value deviates above the norm. So this is what we would refer to as negative feedback. Deviation of a factor to above or below the norm initiates a reflex activity that restores the norm. And this idea of negative feedback, this mechanism, is at the heart of almost all homeostatic mechanisms. So examples of homeostasis include these: control of body temperature, pH of blood and other fluids, the control of osmotic potential of body fluids such as the blood, control of blood glucose levels, control of ion concentrations in the blood, and many, many others. Now, it's our understanding of homeostatic mechanisms which allow us to develop technologies to sustain our lives in very, very harsh environments like these. So we're going to get into a number of different homeostatic mechanisms involving hormones. Now again, we do get into a lot of different hormones and their mechanisms and their glands in a lot more detail within this course module on the website, the course module entitled Control Systems. For now, we're just going to make reference to some of these hormonal mechanisms, but we're not going to go into them in too much depth. So there are two main methods of coordination or control in the body. There's nervous control and there's hormonal control. Now the body requires both of these forms of coordination to be operative at all times and to maintain health and life. Now each type of coordination has its own characteristics and advantages and disadvantages. So the initiation and control of a response will be nervous and or hormonal in nature. Now we can compare those two systems here. So again, I would encourage you guys, if you're watching this as a recording, to pause the video and take those values in. So let's talk a little bit about hormones and coordination here. So a hormone is a substance which is transported by the circulatory system in the blood, and because of this, hormones will reach all parts of the body and all body tissues. However, hormones will only cause effects on specific target organs and tissues which have receptors which are specific to that hormone molecule. So for example, adrenaline only affects the muscles, the heart and the liver, whereas estrogen will only affect the ovaries, the uterus and the pituitary gland. So for example, we have a stimulated endocrine organ. The hormone is released into the bloodstream. The hormone travels around the circulatory system and ultimately is delivered to all the tissues and organs of the body, but only specific target organs will have cells that contain the receptors on their surfaces for that hormone, and therefore will be affected by that hormone. So the main endocrine organs are shown here in these two diagrams, and obviously some hormones have specific effects in females, such as causing the ovaries to release the ova, the uterus to contract during birth, and the mammary glands to produce milk. But some hormones will only have specific effects in males, such as stimulating the growth of skeletal muscle and the testes to produce sperm. So this table again summarizes some of the features of the most important hormones. So I'll let you guys take a quick look at that. Losing my voice, so I'm going to take another sip of my tea. Okay, so let's talk a little bit about the specific hormones and homeostatic mechanisms which are mentioned in the specification here. So let's start with the control of blood glucose here. So the maintenance of a suitable level of blood glucose is necessary for good general health. Now glucose is the main energy source for respiration, and is actually the only energy source that can be utilized by brain and red blood cells. Now normally blood glucose levels remain remarkably stable regardless of how much glucose you ingest or how little you ingest. However, if the control of blood glucose sugar levels fails, then the person will suffer from a disease known as diabetes. Now before the discovery and the isolation of insulin, diabetes was inevitably fatal. And it was for this reason that this gentleman here, Frederick Banting, and also another gentleman named John MacLeod, they were awarded the Nobel Prize in 1923 for the discovery and the isolation of the hormone insulin. Now, Banting and MacLeod shared the prize money with two others, Best and Collip, who had both played important parts in that discovery. Now, this breakthrough was a revolution in medicine, and it gave hope and life to sufferers of diabetes. So, to put this into context, 15 million people living today would have died of diabetes in early childhood had it not been for the discovery of insulin. So we now know that there are two hormones released by the pancreas, insulin and glucagon, which are responsible for keeping the blood glucose level constant by homeostatic mechanisms. So we said before that glucose is the most common respiratory substrate utilized by cells in general, and in fact brain cells and red blood cells can only use glucose as a source of energy. So in normal circumstances, the levels of glucose in the blood remain stable under homeostatic control of those two hormones. But many other body tissues can use fatty acids as a source of metabolic energy in addition to or instead of glucose. But again, the brain and red blood cells can only use glucose. Now, if you're deprived of glucose, then brain disturbances will rapidly follow that. Now again, red blood cells actually lack mitochondria, and they can only obtain their glucose by anaerobic glycolysis. So the glucose that circulates in the bloodstream is derived from three main sources. Now there's obviously the dietary intake and the digestion of carbohydrates, but there's also glycogenolysis, which is the breakdown of stored glycogen, which is a complex carbohydrate found in muscle cells and liver cells. The breakdown of glycogen to form glucose is called glycogenolysis. But you also have another process called gluconeogenesis. Now this is the process by which non-carbohydrate sources such as amino acids can be converted into glucose, typically under conditions of starvation. So dietary carbohydrates include sugars, starch, cellulose, and during digestion disaccharide sugars such as maltose and starch are hydrolyzed to ultimately yield the monosaccharide glucose. Now we get into the structures and more detail about these molecules in the course module entitled Biological Molecules on my website, but also if you want more detail on the structures of these macromolecules and their monomers, you can have a look at the revision webinar that I did on the subject of biological molecules. Now I realize that I haven't yet uploaded that to the YouTube page, but that will be coming soon. I'm just having a few problems with editing that video, so that should be up there fairly soon. Now, as we said before, glycogenolysis is the breakdown of glycogen, which is found in liver and muscle tissue. This can be used to release glucose into the bloodstream if you are lacking glucose in your diet. But you can also convert things like amino acids into glucose by the process of gluconeogenesis. So blood glucose levels, as we said before, are controlled by two principal hormones, both of which are released by the pancreas. These are glucagon and insulin. Now the pancreas is predominantly an exocrine gland. Now the term exocrine refers to glands which release fluids or substances into body compartments other than the bloodstream. But the pancreas also has an endocrine component. Now the pancreas contains clusters of endocrine cells called the islets of Langerhans, and it's these cells which secrete those hormones, insulin and glucagon, directly into the bloodstream. Now the concentration of glucose in the blood in healthy individuals normally lies somewhere between 900 and 100 milligrams per 100 centimeters cubed of blood, and this equates to a molar concentration of 5 to 5.6 millimoles per litre or per decimeter cubed. Now, a rise in blood glucose levels above the norm, for example, after a meal, this is detected by the beta cells of the islets of Langerhans, and these respond by secreting the hormone insulin into the blood. Now, if the blood sugar level drops below the norm, so for example between meals or after fasting, then the alpha cells of the islets of Langerhans detect that, and they respond by secreting the hormone glucagon. So this is a transverse section of the pancreas shown at a magnification of 400 times. Here we can see the region called the islets of Langerhans. These are the endocrine cells of the pancreas. And within those that region, we can see the alpha cells, which detect a fall in blood glucose level and again respond by secreting glucagon into the bloodstream. Here we can see the beta cells, which detect a rise in blood glucose concentration and will respond to that by secreting the hormone insulin into the bloodstream. So the alpha and beta cells of the pancreas are therefore receptors because they detect deviations in blood glucose either above or below the norm. So a negative feedback mechanism operates to return blood glucose levels back to the norm. So insulin decreases the level of blood glucose by the following processes. It increases the permeability of body cells to glucose by stimulating the incorporation of additional glucose carriers into their cell membranes. It also triggers increased glycogenesis, so the activation of the liver enzymes that convert glucose into glycogen is also promoted by insulin. So this obviously will have the effect of sequestering glucose from the blood and building it up into glycogen stored in liver and muscle cells. Insulin also decreases the level of blood glucose by triggering an increased rate of lipogenesis. So this is the conversion of glucose into fatty acids in adipose tissues or fat cells. Now glucagon increases the level of blood glucose by firstly increasing the rate of glycogenolysis. So again, the glucagon activates liver enzymes that convert glycogen into glucose. It will also increase the levels of blood glucose by stimulating gluconeogenesis, in other words, the activation of liver enzymes that convert non-carbohydrates, such as amino acids, into glucose. And finally, glucagon also increases the levels of blood glucose by stimulating lipolysis, which is the breakdown of triglycerides into fatty acids and glycerol in adipose tissue, which can then be converted into glucose. So this diagram here summarizes what we've been talking about here. And these are the two different negative feedback mechanisms by which the alpha and the beta cells of the islets of Langerhans in the pancreas control the blood glucose concentration via this dual hormonal action. So again, to contextualize that in the same way that we saw in that previous diagram, we have the normal blood glucose level shown by this range here, the set point, if you like. A rise in blood glucose stimulates the beta cells, and these then secrete insulin, which then will counteract that rise in glucose by decreasing the glucose concentration of the blood by activating or promoting these processes here that we mentioned before. So this has the effect of restoring the concentration of glucose in the blood. So again, this is an example of negative feedback. And in contrast to that, a fall in blood glucose is detected by the alpha cells, which respond by secreting glucagon, which in turn increases the blood glucose concentration via the promotion of these processes, and therefore, again, restores the norm of blood glucose concentration, and again is an example of negative feedback. Now we mentioned that insulin has an effect on the permeability of the cell membranes of cells to glucose. Now the way that it does this is that cells will typically have a glucose carrier which carries glucose across the membrane of cells via facilitated diffusion. So what happens when insulin attaches to its receptor in the surfaces of these cells, it triggers an intracellular chemical signal which then causes these vesicles containing these glucose carriers to fuse with the plasma membrane, and this in turn increases the number of these glucose carriers in the plasma membrane, which in turn serves to cause more glucose to pass into the cells from the blood. So protein hormones like insulin and glucagon, these are polar lipid-insoluble molecules which are unable to diffuse through the lipid bilayer of plasma membranes. Now these hormones actually bind to receptor proteins in the plasma membranes of their target cells and trigger a chain of events that activate or inhibit enzymes required for specific biochemical reactions. Now the hormone itself is referred to as the first messenger, and on binding to the receptor at the surface of the plasma membrane, the hormone activates specific molecules at the membrane on the underside, which lead to the release of a second messenger, and this in turn enters the cytoplasm and triggers a cascade of events which ultimately results in the response. Now, the glucagon second messenger is a small molecule called cyclic AMP, or CAMP for short. And the involvement of two messengers, the hormone and the cyclic AMP, this has the effect of amplifying the original signal. So cyclic AMP is actually a very widely studied second messenger molecule, although other molecules will perform this function for certain other hormones. So again, the mechanism of hormone action: the hormone binds to the cell surface receptor. The binding induces a conformational change in the receptor, or a change in the shape of the receptor. This then activates a protein called a G protein. Now this in turn activates the enzyme adenyl cyclase, which changes ATP into the second messenger molecule, cyclic AMP. Now the cyclic AMP molecule then goes off into the cytoplasm of the cell and can activate certain enzymes which are required for the specific biochemical reactions which can then trigger the response. So when a protein hormone binds to its cell surface receptor, a cascade of events is triggered, with one event leading to another. So each molecule within the cascade system activates many molecules at the next stage, such that there's an amplification of the original message triggered by the hormone. So in this way, a single molecule of hormone can promote the synthesis of thousands of molecules of the final product. So each activated protein activates many molecules of adenyl cyclase. Oh, let me go back. In turn, let me just play this. So in turn, each activated adenyl cyclase molecule converts many molecules of ATP into cyclic AMP. Each cyclic AMP molecule can activate many desired copies of the enzyme, and each enzyme molecule in turn can catalyze the formation of many molecules of product. So in this way, the binding of one hormone molecule at the cell surface membrane promotes the synthesis of many thousands of the second messenger molecules. This is an example of an amplification, and in turn, a small concentration of hormone in the blood can produce a massive response within the target cells and tissues, and this is what we refer to as amplification. So if we're looking at the effect of glucagon on liver cells, binds to the cell surface receptor, activates the G protein activates adenyl cyclase produces many molecules of cyclic AMP, many molecules of cyclic AMP can inactivate enzymes, can activate inactive enzymes such as protein kinase A, and the active enzyme, active protein kinase A, can then begin to activate other enzymes such as phosphorylase enzymes, and this then catalyzes the conversion of glycogen into glucose, for example, and then that glucose can enter the bloodstream. So we need to talk a little bit about diabetes mellitus to finish off here. So a breakdown in the homeostatic mechanisms that normally control blood glucose concentration can lead to this condition. Now, this is characterized by an inability of the cells to take up glucose from the blood. Now, if the diabetes is untreated, the cells are forced to draw upon other sources for energy, such as fat and protein reserves. So in diabetics, when the blood glucose concentration is high and exceeds the ability of the renal system to excrete, then or to reabsorb, then glucose becomes excreted in the urine. Now, diabetes can be caused firstly by the pancreatic beta cells failing to produce sufficient insulin, but it can also be caused if the insulin receptors in the membranes become abnormal and cease to function. So there are two principal types of diabetes mellitus. The first, which is called type 1 or juvenile onset diabetes, this appears suddenly during childhood as a result of the destruction of the insulin producing cells of the pancreas. Now this can be caused by viral infections, but it can also be caused by autoimmune reactions. So type 1 diabetics are insulin dependent. They require an exogenous source of insulin. Now the other type of diabetes is called type 2 and sometimes referred to as maturity onset. Now this is generally a less severe form of the condition in which the insulin levels may be normal or slightly reduced, but in this case it's the target cells failing to respond to the hormone because of abnormalities in the insulin receptor. So, the undersecretion of insulin and the inability of the cells to use glucose as a respiratory substrate leads to a number of metabolic effects and symptoms in the diabetic. These can include hyperglycemia, which is an increase in blood glucose concentration and the excretion of glucose by the kidneys. Now, this is abnormal. In healthy individuals, normally there is no significant level of glucose found in the urine. The other thing that can happen is protein catabolism, the breakdown of muscle proteins to amino acids in response to the inability of the cells to use glucose as a principal respiratory substrate. So the body will respond by undergoing gluconeogenesis by breaking down proteins, and the main reservoir of proteins in the human body is in the muscles, and so generally the muscles tend to get broken down and the excess amino acids are converted to both glucose and urea in the liver, and this in turn increases the excretion of nitrogen and raises blood glucose levels. Another effect is fat catabolism. So again, because of the inability of the cells to utilize glucose, the body fats start to be hydrolyzed to be broken down to fatty acids, and so that the cells can then utilize the fatty acids as a respiratory substrate. So you end up with excessive fatty acid oxidation, and this produces an excess of acetyl-CoA molecules, which are then converted into ketones by the liver. Now, the release of these ketones into the blood then has the effect of lowering the blood pH and making it more acidic. This is called ketoacidosis. Now, another effect is osmotic diuresis. Now, this is where the high concentrations of glucose in the urine creates a hypertonic urine, which reduces the water reabsorption from the collecting ducts, and so under these conditions a large volume of urine is excreted. So again, this diagram here shows the overall effects of the undersecretion of insulin that we were referring to in the previous slide. So we've mentioned these already, and I'm whizzing through these because I'm conscious of we're running out of time here, and we've still got quite a bit to go. So how is diabetes treated? Well, treatment of type 1 diabetes, again, is by subcutaneous injections of insulin. Insulin cannot be taken orally because it's a protein, because of its protein nature. If you were to take this substance orally, it would simply be broken down by the protease and peptidase enzymes in the stomach. So the insulin dose needs to be carefully adjusted because excessive doses of insulin, together with a low carbohydrate intake, will result in hypoglycemia, or a low blood sugar level. So blood glucose levels have to also be regularly monitored by type 1 diabetics to determine just how much insulin they need. So biosensors are used by these individuals to keep track of their sugar levels. Now, in contrast to that, the treatment of type 2 diabetes, well, obesity is actually a significant contributor to the onset of maturity onset diabetes, or type 2 diabetes. So treatment of type 2 diabetes largely involves dietary controls and changes to the diet and the lifestyle. So nutritionists will typically work with sufferers of type 2 diabetes to devise healthy eating plans that limits the amount of sugar intake and is balanced by an appropriate level of exercise. Now, many modern methods of treatment available for these sufferers of type 1 or type 2 diabetes actually enable these patients to lead relatively normal lives despite the otherwise severe condition that they are suffering from. So, in terms of the causes of hypoglycemia, well, a diabetic may experience low blood sugar if, for example, they miss a meal, or too much insulin is injected, they've done too much physical activity. And the symptoms of hypoglycemia are things like sweating, blurred vision, shaking hands, hunger, and an inability to concentrate. And so the sufferer is often described as having a hypo. Now, these symptoms disappear when the sufferer eats foods containing sugars. So for this reason, sufferers should carry chocolate bars or biscuits with them, or have them within easy reach. So an untreated hypoglycemic person can become unconscious, and it can be life-threatening at that point unless they're treated immediately. So what about hyperglycemia? Well, a diabetic may experience hyperglycemia if they've ingested too much sugar, if they've injected too little insulin, or if they've done an insufficient level of physical activity to use up the ingested sugar. And again, the symptoms here of hyperglycemia would be things like nausea, stomach pains, feeling sleepy, and again, an untreated hyperglycemic could become unconscious, and at that point the condition can become so severe that it can become life-threatening. So in order to test for blood sugar levels, type 1 and type 2 diabetics need to be very conscious and very wary to measure their blood sugar levels at certain times of day. Nowadays, the testing kit is very sophisticated and is very easy to use and gives results more or less instantly. So here we can see the applicator, and it's used to push a lancet, a small needle, into a fingertip. This causes a small drop of blood to then allow you to place a small drop of blood onto the sensor, the biosensor here, and then the sugar level is read by the device, and you can see that sugar level displayed on the meter. And then, of course, the diabetic can then go on to either consume sugar or monitor or regulate their sugar intake and their physical activity accordingly. Okay, everyone, it seems like we've reached the end of the third hour of this webinar. We've still got the topic of the structure and function of the kidneys and osmoregulation to cover. Now, what I will do is I will leave it up to you guys. I'm going to give you guys a 5-minute break. I will leave it up to you guys as to whether or not you want to continue, whether you'd like me to continue. If not, then what I will do is, according to what you guys want, I can either upload a video that covers osmoregulation to the YouTube channel. Alternatively, I can reschedule an additional part 2 to this webinar where you guys can attend and we can go through osmoregulation. I see that some of you are already telling me what you'd prefer. Let's have a look here. How long will it take? Well, I would say that probably to cover what we need to cover in detail, probably maybe just under another hour. So it's up to you guys. I mean, I'm going to, because we've hit the 3-hour limit, in keeping with the kind of format of all of the webinars that have gone before, I will be shutting off the YouTube live stream shortly, and we will then get into the Q&A, and I will answer the questions that you guys have asked in the Q&A box so far. Okay, it seems like most of you are saying for me to upload a video. Okay, so what I'll do is I will shut off the YouTube live stream shortly, and I will get into the Q&A with those of you who are in attendance in the live Zoom room. Probably this evening I will upload an additional video to the YouTube channel to cover the kidneys, the structure and their function, and osmoregulation. And at that point, if you have any questions on that section of this topic, you can either type them in the comments on that YouTube video. Now, alternatively, you can always go to the lesson in the control systems course module. I know many of you have already enrolled on that, and you have access to that lesson. That lesson contains a full teaching video on this topic as well. So I will upload that video to the YouTube page as well. And obviously, those of you who have access to that, you also have a private coaching area where you can type any questions relating to that topic for me to answer as well. Okay, everyone. Well, I will stop the webinar there or at least stop the YouTube live stream, and I will get into the Q&A. But I will give you guys a 5-minute break before I do that. But to those of you watching via the YouTube live stream or watching as a recording, I hope that was useful to you. The next webinar, let me just bring that up on the screen here. So the next webinar in this sequence will be tomorrow at the same time at 11 AM. We're going to get into the A2 topic on genetics, population, evolution and ecosystems. So hopefully I will see you then. Again, if you have any questions, by all means post those either on the lesson pages of my website, invisible-college.com. Alternatively, you can always ask those questions on the YouTube channel on the relevant lesson videos. So again, I will see you in the next video, guys. Take care.